Okay, I think we'll go ahead and get started. We do have a very packed agenda, so we want to stay on time. Um, I'll just introduce myself for those of you who don't know me. I'm Dr. Stephanie Smith. I am the Divisional Director and Section Chief for Infectious Diseases here at the University of Alberta and in the Edmonton Zone. And it is my pleasure to welcome you to our third annual ID Research Day and our fourth annual Dr. Jeff Taylor Lectureship. I will start with just a little bit of an agenda for the day, and then we'll get right into the program. I will also just I'd like to welcome the international academic students that are here from China today, who are in the room, and also those who are online. So I will start with our territorial land acknowledgement. I respectfully acknowledge that we are located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories and languages and cultures continue to enrich our vibrant community. Okay, so the agenda for the day, I think most of you received kind of the order of business, but we do have two invited Research Day lectures, as well as our Jeff Taylor Lectureship, which will take place kind of at the end of our ID Research Day. And then we have our students who are going to be presenting their work. And this is really, I think, the focus of our research day is to really acknowledge and highlight the amazing work that all of our students have done over the past year. And I think it's amazing to see the breadth of research that's done in our division. We have everything from basic science to epidemiologic research to QI. So I think it will be really interesting to hear their research and really to, acknowledge just the amount of hard work that goes into these projects. So I will start off our agenda by giving our Barb Romanowski Award winner. So Dr. Barb Romanowski was a divisional member who was a pioneer in research in STI. And this award was established in her name and is awarded to a publication within sexually transmitted infections. So this year, it's my pleasure to announce the winner is Abhishek Shinde, who is a student of Dr. Joel Dax, and the award is given for his paper, "The Retromer and Retriever Systems are Conserved and Differentially Expanded in Parabasalids." And so this is related to, for those of you who maybe are not as familiar with this basic research, it's related to Trichomonas vaginalis. Please put your hands together for Dr. Shinde. He's not here in the room. I believe he is online. Dr. Shinde, can you tell us if you're here and able to?
Hello.
Hello.
Hi, Dr. Stephanie. Thank you so much, and I can hear you. And yeah, firstly, I would like to correct myself and say that I'm not a doctor yet. I'm still a PhD researcher. So thank you so much for that title, but I still have a long way to go. And yeah, thank you so much for the award. This was very gratifying, and I feel really great about it. Thank you.
Thanks very much. Okay, so I will now invite Dr. Joel Dax to come up. He is the co-chair of our research committee, and he will introduce our first speaker.
Great. Morning, everybody. Okay, so it's my pleasure this morning to introduce Dr. Simon Otto. Dr. Otto got a PhD from the University of Guelph, a DVM from the University of Saskatchewan, and originally hails from Calgary, but we won't hold that against him. He's an associate professor at the University of Alberta School of Public Health and is the lead for the HEAT AMR, so that's the Human Environment Animal Transdisciplinary Antimicrobial Resistance Research Group. He's a veterinarian and an epidemiologist whose research investigates the One Health epidemiology of AMR. HEAT AMR focuses on integrated antimicrobial use and resistance surveillance, diagnostic testing, and stakeholder engagement using quantitative and qualitative approaches to inform and support realistic strategies for antimicrobial stewardship using a One Health approach. He's also the thematic lead, area lead for healthy environments in the Center for Healthy Communities at the School of Public Health. He served on the Canadian Academy of Health Sciences expert research panel on AMR and AMU in food-producing animals. He's a member of the Public Health Agency of Canada Expert AMR Advisory Group for the PHAC AMR Task Force and the Prairie Hub lead of the CIHR-funded Canadian AMR Network. So I'm very excited to hear your work. Here is, there you go.
Good morning, making sure that's coming through. Yes, great. Computer's gone blank. All right, thank you very much, Dr. Dax, Dr. Smith, everyone for coming this morning, for the opportunity to speak. I'm a veterinarian by training. I like to talk about One Health, and I see some friendly faces in the audience, some of which I presented with before. Stan, good to see you this morning. And I want to start, I've been learning from some of my colleagues working with Indigenous partners about territorial acknowledgements, but also situating oneself. And so it's a great start to talk about me, my background, and my relationship with the land as I learn to work with our Indigenous partners. So I am a farm kid. I grew up on a farm in southern Alberta, grain farm. I have strong ties and passion for agriculture. I work a lot with animal agriculture, and you'll see some of that in my work. But I want to point out that One Health, you know, we talk about the integration of the health of humans, animals, and the environment, but realizing that it is still a fairly Western-centric colonial term. And as I learned from Indigenous elders and residents in the School of Public Health, I realized that they really embody One Health, but they don't use the label that we use. And kind of really, I'm going to talk today about how we look at the environment and animal health integrating into this complex, wicked problem of AMR. So let's get started. There we go. Just some brief acknowledgements and disclosures, some of which you've heard already, just a list of some of the grants that I've held, collaborations, some of the contracts with industry partners like Alberta Pork, British Columbia Ministry of Agriculture, the ABVMA, et cetera, and some of this work you'll see. And I think we've already talked about this in my introduction, but if you're interested in any of our work, my website's there, and we're going to get into some of these specific pieces today. So I want to talk about why AMR is a One Health issue. We'll talk about some examples of what I think are AMR One Health conundrums and things that we need to be aware of when we look at controlling AMR and promoting stewardship. A little bit about some of the work that we've done evaluating One Health AMR surveillance in Canada, and I'm excited that Dr. Saxinger is here to speak this morning because I worked with her on some of that, and you'll see her name on some of these things, and then some of the other research that we're working on right now. I like the new definition from the WHO website and the quadripartite about One Health being an integrated, unifying approach aiming to sustainably balance and optimize the health of peoples, animals, and ecosystems. And the reason that I bring this up is it is a transition. The previous definitions used to be really focused on how the environment and animal health contributed to human health, but this is an important transition that it's not just about the health of people. It's about the health of all three of these sectors and about the fact that they're all linked together. So when we make changes in one, we're potentially impacting, cascading down into impacts in other things. And that we can't just be focused on the health of humans because all of these things really tie together quite closely. And if you look at the One Health Joint Plan of Action coming from the quadripartite, you can see that Action Track 5 is specifically focused on antimicrobial resistance. There's obviously important pieces within all these action tracks, but I just wanted to highlight that at the international level. So at the risk of being too simplistic, and I realize we're going to hear from Dr. Huang today about MDROs, but just setting the stage and a reminder that an antimicrobial is any substance that kills or slows the growth of bacteria or microorganisms, but causes little or no damage to the host. And so obviously I'm focused a lot on bacterial diseases, but recognizing that really antibiotics and antimicrobials are talking about antibacterials, antivirals, antifungals, antiparasitics. And while I cannot claim expertise in those bottom three, they are important. And as we look more into resistance in those spaces, we're realizing that that's a problem as well. It's not just the bacterial diseases. It's probably partly a fact that we know more about AMR in the antibacterial space. It then is important to give you a sense of how we use these drugs in animal agriculture and how it looks a little bit different sometimes from how we use these in human medicine. We use them in therapy, so we will treat individual animals that are sick, but we also treat groups of animals that are sick. If we have a broiler chicken barn with 10,000 broiler chicks in it, and there's an infectious disease going through there, usually we need to treat the whole barn. And that means that we're treating some animals that aren't sick, but we have two big barriers. We don't have a diagnostic test that we can tell within minutes which birds need to be treated and which ones don't. We don't have the ability to do that. And the stress we would cause of trying to handle those birds and figure out who's sick and who isn't is going to cause more problems. So we do tend to treat groups of food animals. We use these drugs prophylactically, and we have this wonderful term in food animal production called metaphylaxis. They distinguish the two. They're similar, but the idea that we also are preventing disease by using antimicrobials in high-risk animals. The idea of metaphylaxis is, in particular when we're treating a group, is that we know that some animals within that group are already sick or are going to be sick. So if we treat them, we are going to prevent disease or treat disease in those groups. The one that's a little less nice to talk about is growth promotion. So we have had antimicrobials approved for growth promotion in food animals in Canada for decades. The idea that we can give long-term low-dose antimicrobials to these animals and they will grow faster and more efficiently. We made changes in Canada in the last 6 years to say we are no longer allowed to use category 1, 2, and 3 medically important antimicrobials for growth promotion in food animals. There's some nuance there to how that's actually rolling out. But we really are trying to tackle this because of the potential selection pressure where we're giving these low-dose antimicrobials to groups of animals for long periods of time. Obviously, it's unique that we're treating animals at the group level. We don't go into a daycare when kids come to daycare in the fall and treat them all with antimicrobials because of respiratory disease that might happen. We don't do that. We do that in feedlot cattle. The bugs are a bit different. There's good reasons for that, and I'll talk a bit more about that in some of the testing research that we're doing. But it definitely looks different when we're looking at these groups of animals. But that means we've got these whole populations of animals and their bacteria and potentially their environments that are exposed to antimicrobials or these resistant organisms. So I'm not going to spend a lot of time here. We know that antimicrobial resistance is when these organisms resist the effects of an antimicrobial drug. We have intrinsic resistance where they are always resistant, usually from a genetic phenomenon. There's some move to change that language to expected resistance phenotype. I won't get in the weeds there. But where we're often focused is this acquired resistance. So these bugs weren't resistant, and then they become resistant by exposure and acquiring resistance genes, either through genetic mutation or the acquisition of genetic elements through horizontal genetic transfer, plasmids, integrative conjugative elements, and other things. And the fact that they can share these things between different genera, between different species of bacteria, and that we can spread them between different sectors like human sectors, animal sectors, and the environment. So it's not a good AMR talk in this space, especially in One Health, if you don't put up this lovely figure. Many of you may have seen it before, affectionately dubbed the confusogram. So our colleagues at the Public Health Agency of Canada, the Canadian Integrated Program for AMR Surveillance, use this to guide their surveillance activities, but trying to understand the potential pathway linkages between all of these sectors: food animal production, companion animals, humans, and then our broader environmental pieces here. So we don't, and you can see these bidirectional areas because either we don't know which direction things move or we know that they can move in both directions. But this point being, things can move from animals to human, or animals to humans and humans to animals. They can move in both directions. And then we need to think more broadly about the environment and recognizing there are things missing here. I've been working with some colleagues a bit in Laval, Carolyn Duchesne and her group, who are really focused on bioaerosols and the aerosol movement of AMR. So either in local environments and hospitals, perhaps local environments outside of intensive farming operations, but also more meta movement across in the biosphere and the atmosphere as well. So I hope that this will signify to you that this is a One Health challenge and that we need to think about all of these sectors and potential pathways. And the research that I'm doing and colleagues are trying to understand bits and pieces of those pathways, realizing that it's complex. So it brings us into this idea of the microbial resistome, this collection of antimicrobial resistance genes and determinants that exist and move between humans, animals, whether it be companion animals like our dogs and cats or our food animals, and then our broad environmental pieces, the air, plants, soil, water. And then also understanding that while we think about antimicrobial use in these ones, there is sometimes antimicrobials used in plants and in environmental areas as well. I like this paper. I realize it's 10 years old, but it's a really good review to talk about these phenomena of co-selection and cross-resistance, which are kind of the molecular underpinnings of why AMR is a One Health issue. So we talk first of all about cross-resistance, and this is the idea that a single genetic mechanism can convey resistance to multiple different antimicrobial compounds. So it could be different antimicrobial drug classes. It could be that we have resistance to antimicrobials as well as heavy metals or perhaps biocides, disinfectants, and the like. And a common mechanism for this are these multi-drug efflux pumps that pump things back out of the microbial cell. As humans, we like to define things as antibiotic, disinfectant, heavy metal, biocide. These organisms don't really care what we call them. They've been developing this microbial warfare for billions of years to survive in their environment, to derive energy sources, to outcompete their neighbor, to kill their neighbor. And so it's logical when we think about it that exposure to any one of these things could upregulate a resistance mechanism, which is going to convey resistance to multiple different compounds. The other piece of this is the co-selection of AMR and our mobile genetic elements. So we get things like plasmids that can harbor resistance genes, perhaps to 3 different compounds or antimicrobials, A, B, and C. And if we expose that organism to just antibiotic A, we're going to select for that resistance plasmid to be maintained and amplified, and thereby amplifying the resistance to multiple different drugs at the same time. So keep these things in mind when we're talking this big meta picture, One Health AMR. So when we think about cross-resistance, this paper was focused on efflux pumps. It's an example. It's an in vitro study, but it's interesting. And they said, you know what? I wonder if there's a connection request on the Apple computer here. I don't, just so you know. This is okay. This is an interesting study that hypothesized that co-exposure of Salmonella typhimurium or E. coli to different antimicrobials at different concentrations, as well as common plant agricultural herbicides, might modulate AMR. So they co-expose these organisms in the lab to 3 different things: glyphosate, which is the active ingredient in Roundup, really common agricultural chemical used in plant agriculture, 2,4-D, common as well, and dicamba. Popped up again, just so you know. I'll keep going. And you can get into the paper, but what was interesting is when they looked at this and looked at concentrations, co-exposure with those herbicides modulated the level of AMR. In some cases, in some drugs, it increased resistance, so there's some suggestion that it's upregulating an efflux pump. In other cases, it decreased the level of AMR. But it's this idea that we need to think more broadly than just about how we use these drugs in practice. We need to think about our agricultural practices and other things that could be co-selecting for AMR. This was another interesting study that said, I wonder what happens if we take the human gut microbiome and expose it to non-antimicrobial drugs. And so they found, after looking at over 1,000 different drugs, that 1 in 4 of these drugs with human targets inhibited the growth of bacteria from the human gut. And it could have antibiotic-like side effects in terms of selecting for AMR. And so we think about these drug classes, right? We're talking about statins, antidepressants, allergy medications, medications that we are taking daily, sometimes for the rest of our lives. So that has a different type of selection pressure than just using a course of antimicrobials for an infection. This is an interesting study, and I like to tell the story about my daughter, who at the time when she was younger, curiosity, said, "Dad, what do you do?" And I was talking to her about AMR and research and viral infections, bacterial infections, and then she was interested in dinosaurs at the time. And she said, "You should look at AMR and dinosaurs." And I was like, that's the next best idea. And you go and look at it and realize people have already done that. This isn't dinosaurs, but it's this paleontological approach. And so in this study, there was a region in the UK that in the late 19th century had a lot of industrial activity, and they went into a lake in the region and they took core sediment samples. And they looked at zinc concentrations to correlate to level of industrial activity. And they went into that and found that there was increased genetic markers for zinc resistance traits as well as antimicrobial resistance traits that were positively correlated with the level of zinc in the sediment. And remembering that, of course, in the 1890s, we didn't know what antibiotics were. We weren't using them. But it just reinforces this idea that environmental things and the things that we put into the environment can modulate resistance from a bacterial or a microorganism standpoint. This study exemplifies that sometimes not everything is connected in the way that we think it is. This is a study done by colleagues at Agriculture and Agri-Food Canada down at the Lethbridge Research Centre. And they were really curious about the beef industry. We use a lot of antimicrobials in the beef industry. We know that. I'll talk about that more in a little bit. But they were focused on enterococcal species to understand the potential for resistance transmission between beef production, where we use antimicrobials, transmitting through to potential enterococcal opportunistic infections in humans like E. faecium and E. faecalis. So they took enterococcal organisms from bovine feces, farm samples, feedlot catch basins, so that's the water that they have to control for manure, effluent, and things, natural water sources, beef processing plants, human clinical, and urban wastewater. And the takeaway from those 6 circles is that the navy blue represents Enterococcus hyrae, which was a common isolate in cattle, seems to be more host adapted. So if you look at the bovine sources, feces and feedlot catch basins, you've got more hyrae, kind of a mixed bag in natural water sources. But when you get down into human, of course, we see faecalis and faecium. Beef processing seems a bit funny. You think, why isn't that cattle? But in a lot of cases, I think this was they're using a lot of water coming from urban environments here that's probably going to be more likely to have human-sourced enterococci. So it was a whole genome sequencing study. And so we can see we've got these beef sources here on the left, human sources on the right in red. Obviously, the hyrae isolates here, which a little bit in urban wastewater, nothing obviously on the human-specific pieces. So we've got clinical non-sterile, clinical sterile, clinical vancomycin-resistant enterococcus. And we can see obviously no hyrae in the human clinical pieces. We've got faecalis and faecium down here at the bottom. But what's really interesting is when you look at the resistance gene complements. So the orange tends to represent tetracycline resistance genes. When we look at these hyrae, we have a preponderance of tetracycline resistance genes, which is not a surprise. We do use a lot of tetracyclines in the feed in particular for control of diseases in feedlot cattle. Compared to resistance genes for beta-lactams and fluoroquinolones in the yellow and purple down here in the human isolates. So this study is really showing that it's not this just big mixing soup that everything's the same and everything. It's not that we don't have elements of things moving back and forth, but some other work that I did as a collaborator looking at the potential for AMR transmission from Enterococcus through the food chain found that there really isn't a lot of evidence for it. It doesn't mean that it doesn't happen, but it's probably not the primary source of infection for humans. So let's transition to AMR surveillance, an area that's near and dear to my heart. I've been working with the Canadian surveillance folks since I was a grad student, going on 20 years now. And I want to set the stage a little bit for where we're at and where we need to go. So if you look at the WHO, they state that the purpose for AMR surveillance is the characterization of resistant pathogens and their distributions in the population. Sounds great, nice and clean and simple, but we're missing important things. What populations are we concerned about? What sectors? Pathogens are not the only important bugs we need to consider. Surveillance is a resource-limited environment, and we have priority pathogens of interest, but we need to understand what's going on in commensals, indicator organisms, and that broader resistome as a whole. And then what's the action piece of this? I'm a big proponent that surveillance includes an action piece, whether it's education or some sort of response, but it's more than just providing data. So what does integration look like in a national surveillance system from my perspective? And this is hinging on the work that I've done with Dr. Saxinger. We need to integrate antimicrobial use and resistance data so that we can understand the selection pressures. And arguably, when you look at the last 10 minutes, we probably need to consider more than just the antibiotics themselves. We need to think about the health sectors that we need. We need practitioner level data in a lot of cases, and we also need population level data. What do the interested parties need from integration? Practitioners are going to have a different need than industry groups, which is going to potentially be different than government and other policymakers like regulatory authorities. What's the purpose of that surveillance system? You're going to hear this come through again and again. We're generating data for action to inform evidence-based policy and programs, but what are we actually wanting to do with those data? Are we educating? Are we regulating? Are we auditing, benchmarking, setting targets? And we're good in Canada at setting up surveillance systems, and we do it bit by bit because that's the way they work, but we don't always necessarily know what the endpoint is, or the endpoint changes once we get there, and then it's maybe less fit for purpose. So to me, integration of resistance and use data from representative One Health sectors is our integrated surveillance program, and we're doing that at a level appropriate to achieve our defined objectives. And so then we start to ask this question, what's the role of the environment? And that's where we're going to go for a little bit now. Key Canadian programs, I won't spend a ton of time here because we could talk for half an hour just about this. Canadian Antimicrobial Resistance Surveillance System is that core integrating entity that came about after the 2014 Auditor General's report about Canada's AMR response. And they have largely been data integration. They're taking over some of the primary surveillance responsibilities for some of the new initiatives for communities, long-term care, rural, remote, and northern Indigenous communities. The Canadian Nosocomial Infection Surveillance Program looks at priority pathogens in tertiary care facilities. We've got AMRNet that's starting to feed in antibiogram data, both from human health laboratories as well as animal health laboratories. And then the Canadian Integrated Program for AMR Surveillance. So CPARS, again, could speak for a lot just about this, but this was the only piece, and there's other organism-specific programs that I should highlight in the national scene. CPARS was the only one that was actually designed with a One Health approach in mind. So again, recognizing they're taking human resistance data from across the food chain, sick animals, farm surveillance, abattoirs, retail meat, and they're integrating that down here with antimicrobial use data, which has improved a lot in the last 10 to 15 years, whether it be human clinical and prescription data. We've got animal antimicrobial sales and distribution data, sentinel farm data, and increasingly we've been able to add in use from crops as well as aquaculture. So again, integrating these data over time to understand, but you can see we're still fairly limited in what we cover. Salmonella, Campylobacter, and generic E. coli as an indicator. So this is really aimed at understanding that foodborne transmission. But when we look at this, we could, again, speak a lot about the gaps. So we have wonderful surveillance programs, and they are internationally recognized, but we have big gaps that we need to fill. And the focus of some of this is, for me, has been on the animal health side and the environment piece. So we don't really have much in companion animals beyond MU sales reporting in that collective national distribution data. While we have good representation of food animal commodities, it's still based on a relatively small number of farms. You might have 100 swine farms in that program across Canada, which is a drop in the bucket. A number of the food animal components have been funded through time-limited research, and some of those have been moved to core funding, but there's still gaps in the ability to continue those things. We know very little to nothing about animal pathogens, other than a few things like bovine salmonella and some bovine respiratory disease things. Not to undermine the importance of these things, but again, big gaps in rural communities, community hospitals, long-term care. And then this environment piece lumped down here in one bullet, but the environment is potentially huge. This paper talked about knowledge gaps and understanding of the AMR in Canada, a big list of things, but the part I want to focus on is that the environment is a common theme. Things like how do we do risk assessment from environmental AMR, impacts of heavy metals, cleaning agents, and the like, understanding of resistance in different pieces, and I can tell you that the bioaerosol piece wasn't even on the radar when this paper came out. It wasn't even something we were thinking about. So transition to the current policy environment. Office of the Auditor General released their updated report on what Canada has done in 2023, a focus on multiple things, but on the surveillance side, identifying that we need to improve antimicrobial use and resistance data across all sectors, but in particular in the environment. And then we transition to the Pan-Canadian Action Plan on AMR, released at about the same time, that says, yes, we're going to work on environmental surveillance. And the action plan talks a lot about wastewater, and I'll talk about that more in a minute. But more recently, PHAC has come out with their environmental surveillance strategic framework, which I appreciate, identifying environmental AMR sources and exposure pathways, monitoring spatiotemporal patterns, and hopefully having timely and accessible data to inform action. This dovetails with the recent release of the One Health AMR Framework for Action. Good to see Dr. Jaffe in the room, knowing that this piece, I will laughingly tell this story that Alberta started working on this when I used to work for government, because I was part of that project team. And then the Pan-Canadian Action Plan came, and we had a bit of a side joke and bet, wondering which one was going to get released first. And not to undermine Alberta's, it just, these things take a lot of time, right? And there's a lot of energy, and I'm glad to see them moving forward. So environmental AMR surveillance, what do we actually need to do in this space? And I take us back to one of the important questions is, what is the purpose of that surveillance? What are we trying to do with it? There's obviously other things, methods, users, all of those, but I want to focus on the fact that this purpose of monitoring is not new. Not to spend a ton of time here, when I was with government, I co-chaired a committee for the Canadian Council of Chief Veterinary Officers that said, we need you to tell us how to do antimicrobial use surveillance in animals, because we need to be able to answer questions about how much of this category 1 drug are we using, because we couldn't answer that question at the time. And the challenge is nobody knew what the objective was. So we said, well, you've got sales data, you've got practitioner data, or you've got farm level data. The level of difficulty goes up with that. What question do you need to answer? Because that's going to dictate which one of these you may need to use, or perhaps a blend of them. So what are the objectives for environmental AMR surveillance? If we don't do this, we're stuck with this monitor everything everywhere, which is impossible, especially in a resource-limited environment. So the objectives of environmental AMR surveillance need to consider the fact that links to environmental interventions are not direct. We know that interventions in the human and animal stewardship space are difficult to see outcomes, but if we start trying to link interventions to environmental pieces, it's even more tricky. And what purpose should that surveillance fulfill? Wastewater is great, but that's not environmental AMR. That's telling us largely about human inputs into the environment. Specifically, that's important, but it's only one component. That's fundamentally different than assessing the risk to human or animal health from AMR in the environment. So the environmental role needs to consider 2 AMR-related processes. The fact that we're disseminating resistant organisms and determinants, as well as sometimes antimicrobial residues into the environment. So there's the dissemination and spread. And then, of course, there's the evolution of those things in that environment. I can't remember the number. Joel maybe could tell us. We can only culture something like 15 or 20% of the organisms that exist in soil, for example. So pardon? 1 to 2%. Okay, yeah, bad estimation, sorry. So, there things like metagenomics for the resistome are going to help us start to tackle these things, but we need to consider that. So our motivation for environmental AMR surveillance needs to be linked to one or both of those processes. That paper talks about 5 broad objectives. One is to curb the spread of resistant organisms. So determine the major sources for release into the environment. And these are just schematics to make us think about things like intensive agriculture, perhaps our hospital effluents, wastewater treatment. And as I mentioned, we do use antimicrobials in crops. If you're interested, my colleague Norm Newman has done a lot of interesting work on wastewater adapted E. coli. So these are E. coli that exist in wastewater treatment plants that resist the killing effects of all of the wastewater treatment things that we throw at them, ozonation, UV, heat. So that in of itself is one thing. They harbor a lot of AMR genes. They're not phenotypically resistant, but further work that his group has done has found very close genetic relationships between those environmental E. coli and urinary pathogenic E. coli or bloodborne E. coli, for example, that start to raise questions about, are some of those things surviving the wastewater treatment process? So then we need to understand our exposure from the environment. What are we exposed to from food, from recreational water, perhaps the water that we're drinking after wastewater treatment, and then this bioaerosols piece. That paper does a great job of outlining things, but again, is human-centric. What about our animal exposure from the environment, from all of these things, realizing their feed sources are different, but there's some interesting work looking at antimicrobial resistance in pastures where we've spread either animal manure or perhaps where we've spread solids from human wastewater treatment and the potential for resistance transmission. There's a whole list of things here which I won't spend a ton of time talking about, but environmental contamination. It could be resistant organisms from any or all possible sources, could be pharmaceutical manufacturing, and as I mentioned, we do have to spread solids from human waste, sewage sludge, urine and wastewater, terrestrial animal waste, aquaculture animal waste. You'll hear a bit more about that at the end of my talk. And as I said, we need to think more than just antimicrobial compounds. Perhaps we're screening for changes in AMR abundance, either resistant organisms, if we're organism specific, or maybe we're looking at antimicrobial resistance genes of the entire resistome of all the organisms there. Now, obviously, the methods are going to differ depending on what we're doing, culture-based, whole genome sequencing, perhaps metagenomic sequencing, where we're looking at everything that's in that environment. But how do we then determine what thresholds are, right? What's high, what's low, what's changing? And again, the emerging threats piece is important. We know what we know, but we also know there's a lot of things that we don't know. So our antimicrobial resistance gene databases for genomics work tend to be biased in the genes that we report from the organisms that we study most often. So they're important, we use them a lot, but they're not necessarily indicative of the frequencies and amount of everything that's out there. So do we have the mechanisms and the methods to detect latent resistance genes that either aren't active yet or things that we didn't know about? And what are we looking at? Pathogens, indicator organisms, environmental organisms. And then what do these emerging threats mean in terms of actual risks to human health and animal health? Lots of big questions and areas for research. So I've mentioned Canada's Pan-Canadian Action Plan and the transition of this surveillance data to risk and policy. So PCAP is focused on environmental surveillance, and we have this environmental surveillance strategic framework. Wastewater is good. What actions are we going to enable with that? But what else do we need to do? And then the piece that I didn't write on this slide is we still need to go back and look at the fact that we only have a small number of human pathogens under surveillance. We have almost no animal pathogens. We still have gaps in coverage and representativeness in the human and animal health space. So the environment is sexy from a government funding standpoint, but that's making the assumption that everything else is great, and it's not. There's gaps there. So this is, I mentioned feedlot a few times, and I want to spend a little bit of time here. This is some really fun work that's been funded through Genome Canada, Genome Alberta, and the government of Saskatchewan and Alberta. And so this is a collaborative project that I've co-led with Dr. Cheryl Waldner at the University of Saskatchewan. Really, we've worked a lot with the beef industry, and the beef industry's been proactive, like all of them, about antimicrobial stewardship and doing research. So we know that they use a lot of antimicrobials, just like animal agriculture does. They do it because diseases are a problem. So these are our lovely feedlot calves in the fall. So anybody who's not familiar with feedlot production, we have something like 59,000 cow-calf farms in Canada that range in size from 20 to hundreds of cow-calf pairs. So these farms in the fall ship their young calves and they end up in the feedlot. So they take these calves, it's kind of like putting kids into daycare for the first time. You take them, you run them through an auction mart, you wean them from their mums, you stress them out, and then you throw them into a feedlot where they're already stressed and they're shedding all their respiratory bacteria and viruses around. So bovine respiratory disease has a peak incidence probably in the first 1 week to a month in the feedlot when most of those animals enter feedlots in Canada and Western Canada in the fall. It's the biggest cause of morbidity and mortality. It's a complicated mix of bacterial and viral infections, a lot of bacterial infections, more so than what we see in kids. That's why we use a lot of antimicrobials. These veterinarians also have some of the best data in the world, so they know they need to treat these animals metaphylactically on arrival. Those animals come through the chute. They have a clinical risk assessment based on their history, and they say you're going to get one of probably 3 different antimicrobials for metaphylaxis at the time of arrival, mostly macrolides, some tetracyclines. And they do that because if they don't, way more animals get sick and die. And then they have animals that get pulled for treatment and need antimicrobial therapy. From a diagnostic testing standpoint, in the human health side, we know we would love point of care rapid testing, minutes to hours. We need to make decisions for those animals in the chute in 15 seconds. If we don't have a test result in 15 seconds, it's not going to help us determine what metaphylactic drug they need. And then we need to think about the fact that we're raising these animals in pens, right? So the premise of this study was, can we, first of all, figure out how to take a representative sample from a pen of 200 feedlot cattle, because we can't test them all, can't afford to, take a respiratory sample from their deep nasopharynx, ship it to the lab, do long-read metagenomic sequencing, and then use bioinformatic processing to handle large numbers of samples and data to provide us with the ability to detect common BRD pathogens and antimicrobial resistance genes. And so this paper here was our protocol where we have to use enrichment because we could not get enough sequencing depth to get pathogens and resistance genes over top of the host DNA there from the cattle themselves. So I think we're enriching these for about 10 hours at the lab. So it's not as rapid, it's not chute side, but we can get pen level data now on pathogen and gene detection that will inform treatment protocols for these veterinarians so they can say, all right, if we can get data quickly enough, can you use that to tailor your treatment protocols for the animals that get sick so we're not just using our clinically choice antimicrobials. It's not individual animal medicine, we're not there yet, but it's been really cool because with the protocols and the bioinformatics we're using, anybody who's familiar with the space knows when you do metagenomics and you run your genetic data through the pipeline, you can say in this sample, we've got these organisms and we've got these resistance genes, but the ability to link resistance genes to organisms is tricky. With our long-read sequencing, we know that we can actually identify single reads that say this is Pastorella multocida, this is Mannheimia hemolytica, and on that read we have 1, 2, 3 antimicrobial resistance genes. Integrative conjugative elements are a big deal in BRD pathogens for moving resistance genes around. We have genetic markers where we can detect ICE elements too. So it's really cool the work that we're doing in this space. These other papers here focus on some of the qualitative engagement work that we've done. So the first one, we actually did an exercise in information quality value stream mapping. I'll leave it to you to look that up if you're interested. But it's trying to understand where in the BRD information flow for feedlots could we integrate diagnostic testing to help them understand and manage BRD. And then my PhD student who finished recently just published a paper on the engagement work we did where we interviewed these feedlot veterinarians to understand how sampling works. they've worked with surveillance programs for a long time to collect samples, and we have integrated some of the genetic work we did in field testing with them. But what are the opportunities for testing? What are the barriers? How do we actually generate uptake in that space of such a metagenomic tool? Okay, I said I was going to talk about aquaculture. So another stream of my research is working with salmon producers in BC. So anybody who might follow this will know that we produce salmon in pens such as this. I'll come back to that. So just highlighting here, I've got three master's students actually that have worked on this. Etienne, Jacob, I don't think Rebecca's on this one, but she's on the next one. So we produce salmon on the coast of BC. We do it on the East Coast as well, but we produce salmon in these net pens. Global aquaculture produces more than half of the world's fish supply in the food chain, and salmon makes up the largest portion of Canadian finfish aquaculture. BC is the largest Canadian producer, again, mostly Atlantic salmon. There is public pressure against this. Public generally doesn't like this, but we produce a lot of food. This is done around the world. This is the most highly regulated food producing industry in Canada. They're regulated federally, provincially. I can go online today and tell you which farm site this is, which company it is, what diseases they had, what drugs they used last year. That's the only food animal commodity in Canada where that's done because they are regulated to provide that information, and they do. There's concerns about these fish getting out, intermingling with wild populations, spreading disease. The risk assessments to date show that the risk is minimal because these are as close to domesticated as they get. They know how to eat the food that they're fed, so when they get out, they tend to die because they can't survive on their own. The risks are low, but there is environmental concerns about the manure that settles down to benthic layers from them. We know that that's there. Any intensive agriculture has manure we have to deal with, just like we have to deal with human feces and sewage. I'm not saying that to justify this, but they literally are monitoring benthic layer samples in between downtimes when they have fish there. So I'm trying to set the stage that while not perfect, no agriculture is, they're doing a lot of things to mitigate the risks. So there's about 75 farms located in remote coastal areas in BC. They feed these fish, and yes, sometimes they do have to feed them antimicrobials. They use a lot of vaccinations, and there's only 4 drugs approved in Canada. So just giving you a bit of a background on what this looks like. So our interest was looking at historical surveillance data from the BC government for antimicrobial resistance from their animal health center for the bacterial isolates. So I think it was in about 2015. They used to submit voluntarily, but then federal surveillance required them to submit specimens for bacterial testing starting in 2015. So we had annual prescription data at the provincial level from the feed mills for antimicrobial use data, and then we had this collection of resistance data for those isolates. So I'll be the first to tell you we got the susceptible versus resistant isolate data. We didn't have a lot of information about how testing was done. We don't have genomes or any of those things, unfortunately. But we were able to just look at associations between antimicrobial use annually and antimicrobial resistance annually, just to see if there was linkages at the provincial level. So Etienne did logistic regression modeling with resistance as the outcome. I'll get back to that in a minute, but if you look over this time period, the majority of isolates are coming from Atlantic salmon production, just because that is probably 90 to 95% of the production over that time. And if we look at the bacterial isolates as a whole, there was 65 different bacterial species there, 90% of which I've never heard of before. We could talk more detail if you have questions, but if we look at AMR over time for these drugs, fluorphenicol, oxytetracycline, and the potentiated sulfonamides, which are the approved drugs, and then they did triple sulfa resistance, you can see that the resistance generally isn't high anywhere, some fluctuations up and down. Obviously, the triple sulfa seems to be the highest. Just to give you an idea of the annual bits. And I should have overlaid the AMU on that. I'm sorry, I've got to figure that in a different talk. But so we took those Atlantic salmon isolates because of the relatively small number. We did a model with all of them together, which is not ideal, but we tried to control for different organism species using random effects. So we had a model for potentiated sulfonamide resistance, one for oxytetracycline resistance, one for fluorphenicol. And we found that the biggest, strongest predictor of resistance to one drug was resistance to the other drugs. So there's some linkage there. And the only one where there was a slight linkage between use and resistance was the potentiated sulfonamide use with that resistance. Generally, antimicrobial use was not associated with resistance at all. Again, this is a high-level ecologic study, but these are the data that we have. So AMU generally was not linked to AMR in our models. Resistance to different drugs seem to be linked together, but we are still really trying to understand what are alternative drivers for AMR in this space. So the other piece that Etienne and my student Rebecca worked on was this lovely bacteria, or this disease called yellow mouth caused by an organism, Tenacibaculum maritimum. So TMAR is a ubiquitous environmental organism. This is the BRD of salmon. You take salmon from a freshwater environment as smolts. They're about 100 to 500 grams in size, or 100 grams in size, and you put them at sea after a transition into salt water. And if they're going to get sick from TMAR, they tend to get sick fairly early on. Stress, they move, and it's an organism that lives in that saline environment. According to the industry, this is the biggest reason now for antimicrobial use in BC is to treat TMAR infections. So if they can figure out how to control that and reduce it, they're going to reduce their need for antimicrobials. So we had this data set from an industry partner with all sorts of environmental, other risk factor data, trying to understand risk factors for yellowmouth incidents. So Rebecca looked at overall antimicrobial use in pen placements. Etienne looked at time to first need for antimicrobials, and that's what I'll talk about here, trying to understand linkages. So our outcome was first instance of antimicrobial use as a proxy for TMAR infection. And Rebecca did a whole complicated analysis where we took 30 or 40 variables and tried to screen them down to the 10 that looked like they were probably most important, using decision tree analyses. And then Etienne looked at the survival analysis piece, which if you've ever done it, it's not easy to do. So you can't just chunk 20 variables in a model and figure it out. There's a lot of assumptions. So we focused on salinity of the water, parts per thousand, temperature of the water at a daily level. The days between 100 and 500 grams are how fast these fish grow. So the faster they grow, the idea is the less likely they are to get sick with TMAR. And then the month of placement to control for seasonality. So we did survival curves and Cox proportional hazards models where we controlled for clustering at the area level because of the spatial linkages. And obviously you're going to have differences in environmental pieces over different spatial areas. So what we can see from these for salinity and for temperature is that in the first kind of 13 to 16 days, differences in salinity and temperature don't have a big impact on the relative outcome of TMAR in these. If they're gonna get it, they're gonna get it. But after that time, the more saline you are, green and red, or trying to remember, yeah, and the differences in temperature here, oh, sorry, this is days between, this is growth rate. The slower they grow, the increased risk they are to be infected with TMAR. This came through in our Cox proportional hazards models comparing these tertiles. So the higher levels of salinity, tertile 3 versus 1, 2 versus 1, had higher hazard ratios than the lower. Same thing for days between 100 and 500 grams. So temperature was related, not in this type of relationship, but we knew that salinity, temperature, and growth rate were associated with yellowmouth. Now, unfortunately, these are things that are harder for us necessarily to control, but perhaps there's mechanisms we can think about. And they're really interested, of course, to understand what are the factors now that are going to link to growth rate, obviously. So just winding up, that's been a bit of a troll through One Health AMR, some of the research we're doing. I just want to highlight my amazing team that's done a lot of this work along the way. Thank you very much to all the hard work of students and trainees in the space. And I'd be happy to take any questions for anybody that wants to know. That is deep in Treaty 7 from my parents' farm. So that's the Sweetgrass Hills on the Montana-US border in southern Alberta. Thank you.
So much. Questions? I'll start kind of a nerdy sequencing question. So you're going to be doing long read sequencing was that with minions on site, or was that, I mean, that seemed to be the way you could improve the speed.
We wanted to do that. That was the ideal thought. Can we use Voltrax and the stuff on site? With the initial thought, but when we worked through the product, we couldn't get enough sequencing depth from that initial. We need that enrichment in the lab. So, and this project was funded and we had the money in our pockets about a month before COVID started. So so while we would love to say we have vet clinics doing minion sequencing, in the end, we brought it in. So we ended up doing a lot of kind of automated library prep and sending and doing GridION and Promethean in particular, because we ended up having hundreds, if not thousands of samples. So that was the other, in the field study. I mean, obviously, if you're doing one-offs, it's going to be different. But interestingly, right at the end of this project, we had the opportunity through the sequencing group we used to use PacBio and do a comparison. So it was interesting to see that we were getting comparable results between the two. And so it was actually somewhat agnostic in terms of the long read sequencing platform we were using. But the other piece is that bioinformatics and data management side is huge, right? Like at one point we had half of the U of S's server capacity tied up in sequencing data.
Good use for it.
Yeah.
Question? Mark.
Thanks very much for an excellent talk. The Alberta AMR framework comes with some money and hopefully there will be some environmental monitoring.
Yeah.
Question your thoughts on who you have been thinking about.
Yeah.
Start thinking.
I mean, I could give you where I think it should go, but I don't know that we've had that conversation about which of those 5 objectives we're trying to tackle, right? The wastewater has been the easiest one to start, and I don't deny that that's important. That's telling us important things, but I think, you know, and Mike Parkins at U of C's done some great work in the wastewater space, and they've got research and they're talking about it. And he is now working, I think, on projects to get into wastewater and agriculture, I think, to understand that, because I think we need to start to understand that export to start with. That exposure from is a lot trickier, but that input into the environment is a great first step as long as we're thinking about what's next. Carolyn Deschain's work on bioaerosols is fascinating. I think we're still just starting to understand what's important and where, and we're going to need to figure out how to prioritize some of those pieces. Not a great answer, like I said, but I think the other collective is, what does the industry, government, stakeholders, what does everybody need in that space? So
Maybe one last fast question. Yes, back.
It has been poorly looked at, looking at gender, looking at sex and age. Like I remember the first CIHR grant I tried to write when I started, and I was using surveillance data to look at outcomes of illness, and I got hammered for not looking at gender and sex. And I said, well, I don't have that information. And then the other pieces, they say, well, you know, what about sex of animals? I'm like, okay, yeah, do we need to consider it? But it's the bigger picture of gender impacts of AMRs. We know that AMR impacts different genders, different cultures, you know, in different ways, socioeconomic disadvantaged people. So we need to think about it. The surveillance data is not great in that space. It's probably, hopefully going to get better. How you then take that in an agricultural lens is a whole other ball of wax, but I don't know of anything. And if others do and I'm missing it, please say so.
Great. Thank you so
Thank
much
you.
for your talk.
Good morning, everyone. It gave me great pleasure to introduce our 3 trainee with best abstract submission. These abstracts were submitted for clinical, translational, and basic science. So I'm going to present the 3 of them, and then we'll have a 10-minute presentation for each and 5 minutes for question and certificate with pictures. So don't go away after your presentations. So the first trainee is Veralika Tiagi, and she's doing her master's. And the good news, she is going to start medical school in the fall, and she will be presenting COVID and other respiratory viruses, vaccines and infections in patients that are enrolled in Treat COVID. And then our second trainee is Dr. Joha Ali, who's also doing her master's under the supervision of Dr. Cervera, and she'll be presenting plant-derived extracellular vesicle and their potential role in microbiome and bacterial infections. And the last trainee is Dr. Carmen Say, who's finishing her ID residency, and she'll be joining the group at the Grey Nun for a locum, which is great. And she's going to present her work with the TB group about TB and diabetes in Alberta, Canada.
Okay, good morning, everyone. My name is Verolika. I'm a master's student with Dr. Kabani, and today I'll be sharing a sub-study of the Treat COVID project, focusing on prevention and infection rates of SARS-CoV-2 and other respiratory viruses in solid organ transplant recipients and their caregivers. Transplant recipients are at increased risk of severe disease from respiratory viral infections due to chronic immunosuppression. Vaccine uptake among transplant recipients was initially high during the peak of the pandemic due to public health efforts and heightened risk awareness. However, it is unclear whether vaccination rates have remained high in this population and in their close contacts. Additionally, it is unclear if SARS-CoV-2 continues to cause more severe disease in SOT compared to other circulating respiratory viruses. So the Treat COVID study is a national multi-center study designed to evaluate the clinical, mental health, and economic effects of COVID-19 on transplant recipients, as well as their caregivers. In this sub-study specifically, we aim to characterize the demographic and clinical profiles of participants enrolled in the Treat COVID study, provide information on SARS-CoV-2 vaccinations and infections at time of enrollment, and compare rates of SARS-CoV-2 to other respiratory viruses. Okay, so Treat COVID is an online questionnaire-based study. Participants self-enroll on an online platform through links sent via mass recruitment email or conversations with their clinicians or posters around the hospital. This next slide is highlighting the follow-up timeline of the Treat COVID study. SOT and their family members complete questionnaires every 3 months. At these time points, research coordinators also complete medical chart reviews. If the participant is diagnosed with confirmed COVID-19, the frequency of these questionnaires increases in the short term following diagnosis, and the follow-up period is for up to 2 years. So moving to the results, here's a flow diagram detailing the recruitment across 7 transplant centers. In total, we had 842 participants complete the screening questionnaires and enroll in the study. Pediatric participants and those that did not complete baseline questionnaires were excluded from our analysis. And so in our analysis, we included 527 SOT and 111 of their associated caregivers. On the right is the breakdown of participants by site. This cohort included participants from sites in Ontario, Quebec, BC, and Alberta, with the majority from Quebec and Alberta. Turning to demographic characteristics, the median age of transplant recipients was 59 years and around half were male. For caregivers, they had a median age of 56 years and 30% were male. While there is some racial diversity, the majority of patients or participants were white North American. And as for job status, most participants are either retired or working full-time, with 10% of transplant recipients on disability. Among the enrolled transplant recipients, the organ type distribution was quite diverse. Kidney transplants accounted for 36% of the cohort, followed by liver and lung transplant recipients at around a quarter each. The median time since transplant was 6 years, and around 11% had a previous or retransplant. 2% experienced rejection within the previous 12 months prior to enrolling. The immunosuppressives are also listed here, with 91% of participants taking either one of tacrolimus or cyclosporine, and 66% taking prednisone at enrollment. Moving now to vaccinations. In SOT, 96% had received at least 1 dose of COVID-19 vaccine. 56.8% received a COVID-19 vaccine in 2024-25. 20% did not, and this data is unknown in 24% of the SOT enrolled. For influenza vaccine in the 2024-25 season, 54.8% of transplant recipients received a dose, 22.4% did not, and this is unknown in 23%. Among caregivers, 4.5% remain unvaccinated against COVID-19. 46% have updated COVID-19 vaccine in 2024-25, and 44% have updated influenza vaccine. However, of note is the percent unknown of this variable in both SOT and caregivers is quite high, so that is a gap moving forward that we'd like to fill. Moving to RSV vaccination now, this data is specific to the U of A cohort. In SOT, we had 98 patients which were analyzed. Of these, 48 SOT were eligible for RSV vaccination, and 12 or 25% received it. For caregivers, there were 45 participants analyzed in total. Of these, 29 were eligible for the vaccine and 8 received it. For Evusheld, 114 out of 438 SOT were ineligible for Evusheld, and these criteria for eligibility differed by province and were taken into account. Of note, only 31% or 77 of those eligible for Evusheld received it, and of these, 65% received the full dose of 600 milligrams. Prior to enrolling, 64% of transplant recipients had documented COVID-19. 61% of these had a single infection, 30% with 2 infections, and 8.6% with 3 prior infections. For caregivers, this number is quite similar, with 60% having a previous documented COVID-19 episode. So at the U of A sites, we are providing respiratory swabs to participants and conducting a full respiratory panel if those participants are symptomatic and drop off a swab. So we have observed the following infections in our cohort since enrollment. And these numbers are either from participants who have dropped off the provided swab or have been tested in clinic or hospital. Moving now towards some of the limitations of our study. One of the limitations is the delay in enrollment. So enrollment for our study began in September 2024, and so much of the COVID-19 data included is based on retrospective review and SOT recollection. A second limitation is underrepresentation of respiratory viral infections, including COVID-19. So despite the efforts of non-U of A sites to direct participants to testing kits, there may be limited access to testing for COVID-19 and other respiratory infections for mildly symptomatic SOT. Another limitation is that there are some missing data due to incomplete questionnaires and data that has not yet been collected from medical chart reviews. Lastly, there is some selection bias present. So vaccination data is for participants willing to enroll in a COVID-related study, and so it may not reflect the numbers in the general transplant population. Okay, so to conclude, participants enrolled have a diverse demographic and organ type composition. COVID-19 was common among SOT and caregivers prior to enrollment. SOT had very high vaccination rates, but only around 55% of SOT received a COVID-19 vaccine in 2024. Lastly, non-publicly funded vaccines like the RSV vaccine have less uptake in SOT. So future work will include ongoing enrollment and data completion, analysis of determinants of sustained vaccine uptake, and comparison of COVID-19 versus other respiratory virus incidents and outcomes in 2025-26. I would like to thank all those that supported this work, including the study funders, and thank you all for your attention. I'd be happy to take any questions now.
Like the numbers of patients that have like just 1 year of COVID, 2 events with COVID or more. Like, how are you actually getting the diagnosis and thinking to contact the COVID variant? You know how they got that positive for a very long time? They get like run up into the test or whatever reason they have to go for the time. Or how are you taking that into account to know if it's like
Infection. Okay, so that's a great question. We actually didn't take into account the time between infections or testing of different variants. So perhaps those that had multiple infections were persistently infected.
Kind of one thing, one caregiver tested like using PCR or just self-reported?
Yes, so those are self-reported positive. Yes. Yeah, a lot of them probably didn't have access to the PCR test, so it may be rapid testing as well.
Okay, but so for the caregivers, is it just self-report that they have symptoms and then like how is it determined that they had COVID versus something else?
So either they would have, so this is all self-reported for family members. So this was either they did a rapid test or maybe the SOT was positive, their associated SOT was positive and they developed symptoms, so they self-reported as having COVID previously. Yeah.
I know analysis of factors is kind of right. But is there an inkling, is it more on an education piece, like they're not aware that they're eligible for certain vaccinations versus a hesitancy to some of those vaccinations like RSV that you mentioned?
Yeah, so that's a really great question. I think looking ahead, we'll try to look at different factors that affect vaccination rates and that affect persistent vaccination rates. I think that's as I said, it's a great question, and we'd have to collect more data and look into it a little bit more to perform some of that analysis. Okay, thank you, everyone.
Good morning, everyone. First of all, I'd like to thank the division for this opportunity to present, share our findings on this ongoing project on plant-derived vesicles and their potential role in microbiome and bacterial infections. So currently in the botanical science world, there are mixed messages as to whether starch nanoparticles are a product of chemical modification or they're generated, they're naturally occurring entities. So it's mostly considered that SNPs are generated through chemical modification of starch granules, and they range from 10 to 300 nanometer in sizes, with a chemical composition of amylose and amylopectin. And they've gained much interest in medicine due to their potential application as a drug delivery system. So what prompted us to investigate SNPs was findings of these similar nanoscale particles in urine samples of healthy participants, as well as acidic fluid of patients with liver cirrhosis. And these bacteria-like structures, they shared similar morphology to the described SNPs. As we can see from, these were images obtained from scanning electron microscope. On the right, these were images processed from acidic fluid samples of cirrhotic patients. And we can see there were these cocci-like structures with a size of 200 to 450 nanometers. And on the left, these were images from urine samples of healthy participants. And in these, for most of them, we see like rod-like structures. In some samples, they were entangled in this fibrous mesh. And some samples, we were also able to observe partially disrupted starch granules. So our hypotheses were SNPs are biologically active structures. So a study aimed to characterize these particles across 4 key domains. We wanted to look at their morphology in depth, their DNA composition, cellular uptake, and their functional ability to translocate DNA. So we started by first visualizing them as they emerged out of starch granules. As you can see from this video, this was a cone starch granule, which was fixed. And then immediately when we hydrated them, we could see these nanoscale particles emerging out of the granules. So to characterize the morphology, we visualize them under scanning electron microscope and transmission electron microscope as well. So our findings from scanning electron microscope, we picked these 3 images from these 3 types of starches, although we also analyzed other varieties. And from the lentils and cantaloupe, we can see they shared kind of a similar morphology in size and shape, but the quinoa was unique in that it had this distinctive polyhedral structure. So furthermore, under the transmission electron microscope, what we observed, looking at the image from the left, this was obtained from lentils, and we observed multiple lipid bilayer membrane-bound vesicles with a membrane which had this infolding configuration. And on the right, this was a sample from quinoa, but the membrane configuration here we observed had this extended relaxed appearance. So to further characterize the DNA composition of plant-derived vesicles, the first experiment we did was separating pelleted plant-derived vesicles from the supernatant, which contained free DNA. And after isolating the DNA, we subjected it to 16S RNA gene sequencing to identify bacteria DNA which was associated with the vesicles. So what we observed from this first experiment was that if we look at the chart on the left, we can see that the free DNA had a higher concentration than the pellet. But overall, when the sequencing analysis revealed that the free DNA had very few reads compared to the pellet. And this may suggest that this was probably because the free DNA was highly fragmented DNA. And all in all, the amplified sequences which were recovered from the pellet revealed a majority to be of chloroplast and mitochondrial origin with very few bacterial sequences. So we had another hypothesis that plant mitochondria and chloroplast could be capable of transforming into bacteria. Hence, we did a second experiment to test this hypothesis. And this time, what we did was incubate the vesicles and the free DNA contained in the supernatant together in a period of 24 hours at 37 degrees. And then we proceeded to the same DNA extraction protocol. So the findings from this second experiment showed that 20% of the sequences amplified were chloroplast, while 15% were mitochondria. And the rest of the sequencing reads, which in this experiment now showed there were mitochondria in origin. Bacteria in origin. So these 2 charts give a summary of the 2 experiments. From the chart on experiment 1, as I had mentioned below, majority of the reads were identified as chloroplast and mitochondria, depicted with the blue color code. From the second experiment, it showed the majority of the sequences amplified were bacteria of origin, and predominantly, as you can see color-coded in this orange color, these were bacilli in family. And one important thing to note that bacilli was seen in all the starch types which we analyzed. And when we cultured all this in the process of trying to identify the microbiome associated with the starch, all the plates grew bacillus. So it's highly probable that bacillus is a common contaminant in all these starch granules. So we proceeded further to test if these plant-derived vesicles can be endocytosed. So what we did was pre-stain them with primary antibodies against pelican, which is a heparan sulfate proteoglycan that we found covers the vesicles membrane. So we used HeLa cells in which we loaded these pre-stained vesicles, incubated them for 15 minutes, and after washing and fixing the cells, we prepared them for immunofluorescence. So visualized under confocal microscope, what we observed, if you look at this composite image, we can see the vesicles were endocytosed by HeLa cells, depicted by this distributed red fluorescence signal, which is seen inside the HeLa cells. Okay. So we then explored if these vesicles had the potential to translocate DNA in HeLa cells. What we did was transform plant-derived vesicles with a non-E. coli DNA and then loaded them into cultured HeLa cells. We had case one, which were cells which were exposed to the transformed vesicles, but not starved. Case two, similar scenario, but the starvation was for 3 days. And we had a control where we did not expose them or starve them. We did not expose them or starve them. So under confocal microscope, after processing the samples, what we observed, the expression of E. coli antigen was much more intense and clearer in case two. And the control was negative. Yeah. So furthermore, we loaded the HeLa cells with vesicles transformed with GFP expressing E. coli DNA. What we wanted to do is analyze with Western blot to depict the detection of to depict GFP protein. So in this case, we got a negative blot, but however, we anticipate that prolongation of starvation would lead to increased expression of GFP, and we'll take this into account in the subsequent experiments. So finally, I'd like to share this live imaging video of HeLa cells. These were cells which were cultured in a glass bottom plate and exposed to vesicles which were transformed with E. coli GFP DNA for 3 days. The major difference here was that we had starved these cells for 4 days before analyzing them. And we can see morphologically, we see these structures resembling bacteria and expressing green fluorescence in the GFP range. So to conclude this, we find so far our preliminary findings indicate SNPs to be naturally occurring entities which share some similarities to plant-derived vesicles morphologically, and they have the potential to translocate bacterial DNA and potentially inducing intracellular bacterial communities. The future direction of this project is that we'll be exploring this phenomena with other cells. Currently, we have a particular interest in looking at similar experiments and getting data to how they'll work in mesothelial cells. And this is to better understand the pathophysiology of spontaneous bacterial peritonitis. And we find with more analysis, we may unpack more knowledge on how these plant-derived vesicles may contribute to bacterial infection and therefore improve interventions, therapeutic interventions. Finally, I'd like to thank everyone who is involved in this project, and a particular acknowledgement to Dr. Greg Tyrell for his valuable insight and knowledge and contribution in this project. Thank you.
Questions so we can stay on time?
Yep.
Thanks for that. I'm curious with what you're describing as, so you're saying that if the starch nanoparticles can bring some bacterial DNA, it can create bacteria? Is that what you're?
Yeah, that's the hypothesis. What we're doing is if we hypothesize if we expose the starch nanoparticles, which I mean morphologically have the characteristics of vesicles, they have the ability to uptake the DNA and transform into bacteria. Yeah.
I'm not sure that bringing in DNA creates a bacteria. You're producing a protein, which is fantastic.
Yes.
Like with a transformed plasma.
Yeah.
How you can bring that you can.
Yeah, that's what we're exploring with the experiments we've been doing. And we find that the mechanistic way in how this is happening will be part of something which we can explore in depth in the future.
So I'm just going to dive in. So diabetes and TB pose significant global health challenges, with TB affecting millions worldwide and leading to a significant amount of death that's ongoing. It also continues to persist in Canada at an annual incidence rate of approximately 5.5 per 100,000 population, which has been essentially unchanged for the last several years now. In addition, the prevalence of diabetes is estimated to increase by 50% from 2019 to 2045. And currently, there is limited literature looking at TB and diabetes in high resource, low incidence countries. And so there's currently a bidirectional relationship known with TB and diabetes. And as you may know, TB primarily affects the lungs and then can enter a dormant state termed latent TB. And in the setting of a weakened immune system, such as with diabetes, this can increase the risk of reactivation. And it is also known that patients with both conditions are at an increased risk of having delayed sputum conversion during their TB treatment, having a higher rate of treatment failure and relapse. And then also, currently the association with mortality is a little less clear, with some studies showing an increased rate versus some showing no differences. Currently, the Canadian TB guidelines recommend routine screening for diabetes in those diagnosed with tuberculosis, and those diagnosed should then be linked to a diabetes care provider. However, currently, real-world data on diabetes care linkage, follow-up testing, and transient hyperglycemia in TB patients remain limited in the Canadian context. Additionally, our study looked at serum drug concentrations in this population, and serum drug concentrations are generally done in this population to assess for adequate dosage and levels in the patient. Doing serum drug concentrations in diabetes patients with TB is a conditional recommendation by the Canadian TB guidelines. However, this is based on limited evidence, and it is felt that diabetes can affect drug absorption via multiple mechanisms, potentially through delayed gastric emptying or altered metabolism as well. Lower drug levels may contribute to poorer TB outcomes in this patient population. However, conducting serum drug concentration is very expensive and also time-consuming for both the patient and also the healthcare provider, as it requires generally taking 2 samples throughout the day. So generally 1 to 2 hours after consumption of the drugs, and then approximately 3 to 7 hours depending on the drug again. So the objectives of this study was to identify the predictors of diabetes in TB patients over a 5-year period in Alberta, and then also to evaluate the utilization of serum drug concentration monitoring, And then finally, also determine whether TB patients diagnosed with diabetes subsequently receive specialized diabetic care within the year that they were diagnosed with TB. And this is with referral to either an internist with diabetes specialty or an endocrinologist. This was a retrospective observational study that looked at all cases of TB diagnosed in Alberta between January 1st, 2018 and December 31st, 2022. As you can see, data was extracted from multiple sources, and the diabetes status was determined by review of the patient's medical history, medication use, and their previous A1C values. And then classification of whether they were pre-diabetes, diabetic or non-diabetic was defined by the Diabetes Canada guidelines. Data was analyzed using SPSS statistics software, and when multivariable logistic regression models were used, they were adjusted for potential confounders, including age, sex, corticosteroid use, HIV status, end-stage renal disease, and transplant immunosuppressive therapy. And so on to the results. We had a total of 1,198 patients. One of them was excluded because they died very soon after their TB diagnosis with no A1C value drawn. And 279 of those had diabetes, while 918 did not. Of those 1,197, 1,020 were culture proven diagnoses. And as you can see here, patients with diabetes were more likely to be older, be male, and then also have pulmonary disease as well. And although not shown here, there was no statistical significance in patients regarding diabetes, non-diabetes with HIV and first-line drug resistance. This is a pie chart showing the region of origin for which patients with diabetes come from. And as you can see, there was a statistical significance of which diabetes patients were more likely to be from East Asia and Pacific, and particularly the Philippines. And this was based on the world group country groupings. And then looking at the patients with both TB and diabetes, the mean age was 61 with a mean hemoglobin A1C of 8.6%, and approximately 16% of those were on insulin. And this is looking at the patients who had diabetes. Approximately 17% of those had a termed new diagnosis of diabetes at the time of their TB diagnosis as well. And only 7% of those who had diabetes were referred for specialized diabetes care. And 38% of those who had diabetes had serum drug levels completed. And then following this, we also looked at long-term glycemic outcomes in those who had both pre-diabetes and also new diagnosis of diabetes. And this is just a follow-up A1C measured at any time point between 6 months to 3 years after TB diagnosis. 6 months was chosen due to, that's generally the average duration of patients for treatment for TB. And then so for the pre-diabetes group, we had 59% or 102 out of 173 who had an A1C completed during this time period. And for those who were newly diagnosed, 34 out of 47 or 72%. And this excluded all those who were deceased, absconded, or transferred out. In the pre-diabetes group, only 16.7% progressed to having diabetes. And then in those who were newly diagnosed, 32.4% actually reverted back to being a non-diabetic A1C level without requiring any treatment. And then all of those patients actually had an initial A1C that was lower, so less than 7% at the time of TB diagnosis. And then looking at outcomes, as you can see here, just looking at the deaths, there's actually fairly little deaths, like 72 deaths overall. And when we did the unadjusted analysis, there was a significant difference where patients with diabetes had an increased risk of mortality and also failure. But once we adjusted for the confounders, there was no statistical significance. And then, although not shown here, we also stratified it by A1C levels, and those who had an A1C between 6.5% to 9% actually had also an increased risk of mortality, although again, once adjusted, this was attenuated and no longer significant. And then just very briefly on serum drug concentrations, as you can see here on the second row for isoniazid, rifampin, and rifabutin that were looked at, slightly more than 60% of the patients who had serum drug concentrations level done achieved adequate levels. And then 2 rows below that show that isoniazid and in rifampin, those patients had, a small proportion had what we say delayed gastric absorption with a low peak, but then their second level completed had normal levels. And then finally, once we looked at serum drug concentrations and outcomes, we found that the adjusted analysis showed that serum drug concentration was not significantly associated with mortality reduction in diabetes patients. So in conclusion, we did find from the study that diabetes is common in TB patients in Alberta. So approximately 23.3% had diabetes and 16.8% were new diagnoses. And they were more likely to be older from East Asia and Pacific and had pulmonary TB. And then of note, those without diabetes, there was a proportion that were pre-diabetic, which would be an interesting group to look at in the future for potential interventions prior to developing diabetes, as they may be at increased risk in certain patient populations. And then finally, transient hyperglycemia was also noted in our patient population, with 32.4% of those newly diagnosed reverting back to non-diabetic A1C levels, and all those had A1Cs less than 7%, which supports that in these patient populations, it's important to do repeat testing after treatment completion to ensure a correct diagnosis of diabetes. And then finally, follow-up and care gaps were identified, as only 59% of the pre-diabetes and 72% of the new diabetes had a follow-up A1C test completed, and very few were referred for specialized diabetes care. And a potential intervention would be a referral pathway for TB and diabetes patients, particularly in this patient population where many of these may not have primary care providers. And then serum drug concentration monitoring was noted to be less than 40% in the diabetes population in our group, and that therapeutic levels were achieved inconsistently. But overall, serum drug concentration didn't seem to be associated with mortality. And so its routine use in all patients with diabetes probably require further studying with prospective studies, and that potentially serum drug concentrations done should be more tailored to the specific patient rather than them just having diabetes as a comorbidity. So special thanks to those who helped out along the way and many others not listed. And then I just have some references, and that's it. Yeah. Okay, so I guess the question on chat is, if diabetes and associated delayed gastric emptying has there been evidence that similar challenges exist for clients who are taking drugs such as Ozempic, Wegovy with treatment effectiveness? I'm actually not 100% sure. I think some of these drugs are relatively new, and I'm not sure if they've been actually looked at in this specific scenario. So that's a very good question. I'll have to look into that. I'm not sure if anyone else knows.
I'm introducing our next speaker. My name is Vanessa Meyer-Stevenson. Our next speaker probably needs very little introduction, but I'll go through some of the details in case some of you may not be familiar. But Dr. Lenora Saxinger is an infectious disease physician and professor of medicine at the University of Alberta, recognized for her leadership in science communication and knowledge translation. Her commitment to making complex medical information accessible was sharpened through a 2024 fellowship in journalism and health impact at the Dalai Lama School of Public Health in the University of Toronto. And the focus developed significantly. This focus developed significantly during the COVID-19 pandemic, when she co-led the Alberta Health Services COVID-19 Scientific Advisory Group, authoring over 70 formal evidence syntheses and literature reviews. While immersed in evidence evolution, she took on the public-facing role addressing misinformation, providing more than 6,000 media interviews. Unbelievable, I didn't know that number was that high. Whew. Yep, it's probably, you stopped counting, yeah. And building a Twitter social media following of over 30,000. Dr. Saxonger's academic work centers on science communication, evidence synthesis, antimicrobial and diagnostic stewardship, antimicrobial resistance, and the One Health approach, as she was called out by Dr. Otto's in Dr. Otto's talk this morning. With real world, she supports real world practice improvement as a physician lead with the Physician Learning Program, and recently assumed the role of Director of Science Communication for the Department of Medicine. So please join me in welcoming Dr. Saxonger to the podium.
Thanks very much. I have a lot of talks on misinformation, disinformation, and for today's version, I actually kind of pivoted and took a little bit more of a public healthy approach, as well as some research relevant material. And I mean, I'm going to talk about things that probably seem, as we talk about them, obvious, but aren't necessarily always kind of called out as being obvious in a public domain. And so I really want this to be just an interesting commentary that might spark some different ideas in people listening. So we're going to go over the evolution and scope of the problem of disinformation, misinformation. I'll go over the terminology as well. A little bit of pertinent disinformation science, principles of debunking, and some aspects of a public health approach. I'm going to touch a little bit on the relevance of SciComm and engagement for researchers, and just kind of end with thinking about our role collectively, because it is a jungle out there, but there is a role collectively. And the underpinning concept here is to close the loop. And this is the part where it really kind of ties into a research day discussion. Professor Mark Walport was a UK, he just authored a lot of reports. I met him at one when he was doing a post-COVID report in Canada, and I can't remember which this was from, but these quotes are really good. Science isn't finished until it's communicated. The communication to wider audiences is part of the job of being a scientist. So how you communicate is absolutely vital. And I think underpinning that is the idea that the community pays for the universities, the community supports the universities. Some of that support is waning, honestly. And the kind of final loop back to justify the whole endeavor is not to talk amongst ourselves. It's to tell people what we're doing to help them. So there's a really strong relevance piece, I think, that we lost track of. And part of that might be because in research, we tend to sort of look down on blowing your own horn, at least in some institutions. Other places are better at that. And I think that we also have tended to see engagement in a public discussion in a public domain and social media as being kind of unworthy or not valid. And I think that we really have to challenge those thoughts. The second part of his quotes here, I think, are really important, which is a science-educated electorate promotes ethical thinking and informed decision-making, which in turn greatly benefits the society, environment, and nation's economy. Again, when you put it like that, it seems obvious, but do we actually apply that in our lives? Not necessarily. And I'm also going to point out the dangers. A lot of the stuff I have in this talk are antedate the pandemic quotes and data. So this kind of stuff has been going on for a long time, but I think that we really have a different perspective on it now. So Carl Sagan, love him. The dangers of not thinking clearly are much greater now than ever before. It's not that there's something new in our way of thinking, it's that credulous and confused thinking can be much more lethal in ways that it never was before. So the disinformation crucible is how I kind of think about the pandemic. It really brought a lot of these things that have been percolating along into the public view and into professional view as well. And the infodemic is the other pandemic. So very early on, February 2020, the WHO Director General said, we're not fighting an epidemic, we're fighting an infodemic as well. Like that was very early. That's when people were just kind of wondering if it was relevant to us. But they saw that coming. And I think the scope of it still surprised people. And I remember that was the point at which I decided I was not going to be afraid of taking interview requests because people are going to die of misinformation. And so if I'm going to worry about sounding stupid or having a bad hair day or whatever, like that's very irrelevant to the scope of the problem. And so, you know, you learn fearlessness by doing things. Misinformation actually spreads 6 times faster than accurate information on social media. That is information that antedated the pandemic. false health claims and fake news reach more people than facts, also from like 2018. The top 1% of false news cascades reached between 100,000 and 1,000 and 100,000 people, and the truth maxed out at about 1,000 in one study. And the cure for this problem is very complex. I mean, because there are so many different factors that lead into these things. And one of the things that is a problem is that misinformation causes confusion, causes loss of cohesion. And in a time of increasing polarization and decreasing trust, we actually see establishment of competing realities. US data, there's lots of examples of this, but 33% of people absolutely think COVID vaccines have caused thousands of sudden deaths. Like 1 in 3 people surveyed. Over a third thought birth control makes it hard to get pregnant when you stop using it, because by all means, we need misinformation campaigns against birth control. And those are people who said this is probably or definitely true. And then there's a whole list on the other side. So, I mean, this is extremely commonplace. We have our own bubbles and we associate with our people and we get our news sources. We're not necessarily seeing the scope of what is in the public domain or what people are selecting to see or what they are being selected to see. Now I sound like a conspiracy theorist, but we're going to talk about information bubbles shortly. So you've heard me use misinformation and disinformation. Misinformation is kind of the broader term, so I should probably default to that more, but it's false information shared without malicious intent. And so this is like Uncle Bob sharing a Facebook post about natural immunity being superior to vaccine-associated immunity. Uncle Bob just thinks, oh, that's something people should know, right? He's not doing it to try to undermine anything. Disinformation is deliberately fabricated false information. For example, organized campaigns against vaccines that are designed to erode trust in vaccines and promote adoption of alternate practices, which, by the way, people make a lot of money on. Again, sound like I'm wearing a tinfoil hat, and yet it's true. Fake news is a broader term yet. It's often politically charged, but one thing I want to say about the news is that you can lump in legitimate information or legitimate criticism that is labeled in a dismissive or misleading way. And so that's harder to combat. It doesn't flag as disinformation or misinformation because the actual kernel of it is true, but the framing may actively mislead. And in terms of the scope of the problem, that's actually a bigger piece than, easily labeled disinformation. And the infodemic refers to the overabundance of information, both true and false, that makes truth very hard to find. And so people have to kind of pick and choose what truth they want to take. And that becomes important when you look at the impact of social media. And the scope here is actually measured in lives lost. Like, people die of disinformation. They're still dying of disinformation. Real consequences and real deaths. Measles resurgence. In 2019, people were already flagging that there was an increase of 50% of deaths between 2019 and 2016. And right now, across Canada, with a large proportion in Alberta, we have had more measles cases as of May 17th than we've had in the prior 20 years. Like, what is happening? We're just charging back to the 1800s. I can even get into germ theory fights with people online anytime I want. Like, they don't believe in bacteria. So things have gotten really, really confused. COVID-19 misinformation, it was estimated that over 2,800 deaths from false cures in the first 3 months. So people just taking stuff that they should not take. Vaccine hesitancy is said to be associated with 1.5 million preventable deaths annually, and that number is expected to climb. Vaccines are thought to prevent 2 to 3 million deaths annually as of the pre-COVID era, probably more now. So you can see that a misinformation epidemic can be more deadly than a biological one, ultimately. Because it includes all of them. And it also erodes people's trust in a lot of non-infection associated issues that can lead to bad outcomes, like climate change. And so I also thought this cartoon was quite good. We have the 4 horsemen of the apocalypse. We have war, famine, pestilence, death, and the one holding their phone in front of their face, misinformation. So COVID-19 was an immediate case study in consequences, and social media really showed its chops in terms of misinformation spread. Hydroxychloroquine, like this was a study, I think, from 2020 already. They said there was over 550 cardiac events linked to inappropriate use, but people kept on having to study it because people kept on believing in it. And so at the end of the day, there was like a meta-analysis that showed at least a 10% increase in mortality in patients treated with hydroxychloroquine. So the number of deaths far exceeded that. And it was established pretty early on, but like ivermectin, people kept on having to do study after study after study. It was a big waste of time and energy, and none of that should have happened. Bleach ingestion, water, not bleach, 15% increase in poison control calls. Vaccine hesitancy. There's actually studies that correlated vaccine hesitancy with social media exposure. And mask resistance. You'll see geographic clustering of mask resistance matching online echo chambers. So the virus has biology and misinformation has algorithms. I think a larger effect of the pandemic has been a worsened decline in trust. That sounds awkward, but there has been a decline in trust over time, which started over 10 years ago, but really, really got amped up by the pandemic. And societal trust metrics, there's like a million different sources and they're quite consistent. In this one, scientists and physicians had the highest levels of trust out of everybody, around 80%. That was a decrease from pre-COVID era. Physicians still had the most trust amongst professions. Public health, 64% trust. Traditional media, 42% trust. I'm just going to comment that politicians are dangling down below 20% trust. And so, you know, people in medicine and science still have the podium, but the audience is trickling away. Like, where are they going? This just illustrates that some of these are a bit of a surprise to me, like priests, 27%, bankers, journalists. And I think that speaks to one of the themes that's going to come up, which is who do you have relationships with and who's an abstract concept? We'll get to that. I'm going to point out that disinformation and misinformation is a dedicated machine, and countering disinformation often falls to being a side gig. And so in the pandemic, scientists, physicians, public health were outflanked and outgunned on social media almost immediately. And rearguard action is less effective, and there's data on that. I'm going to point out that 12 people/sites are responsible for 65% of misleading vaccine claims. This is pre-pandemic data. You'll see that children'shealthdefense.org, which was the organization that previously was founded by or led by RFK Jr., yes, was the biggest disinformation source found in this analysis. Bot amplification on social media favors anti-vax tweets, and false news spreads farther, faster, deeper, and more broadly in any study that you can look at. And I have a whole bunch of variations of this quote. I actually had to go looking for the source of this quote because it was 1 in the morning and what else do you do? So I will share that with you shortly. A lie can go around the truth before it's the truth. A lie can go around the world before the truth gets its pants on. And I'm just going to point out that there's a little bit of a paradox in the trust issue, which is people say that they trust physicians for health information and vaccine information, less so. I also think that's interesting because if someone's like, you're trying to kill me with a vaccine, but I've broken my arm, please do surgery on me. But somehow that conflict just kind of keeps on going. 23% of people in this survey said that they seek second opinions from social media. And 37% were influenced by online communities over medical advice. So they're stating trust, but they're looking elsewhere for information, and whether that's an access issue or something else, it's important. So yes, trusted, but certainly not the only voice in the room. So why do smart people, because like there's a lot of these people who are very smart, and actually scarily, there's data suggesting that smart people are better at justifying bizarre beliefs. Like, I'm not kidding. Smart people are better at convoluted reasoning to justify increasingly bizarre beliefs. And I find that very frightening. It's easier to think, oh, you just don't know, or, oh, you're just not that smart. That is not it at all. There's a lot of cognitive vulnerabilities, I think, at play, and there's a little bit more expansion of this later, but confirmation bias, which is we always seek and feel comfortable with information that confirms our existing beliefs. So if you see something that kind of resonates with what you think, you hit share in seconds, You believe it. You're happy someone's talking about it. You'll have an emotional reaction to it. You'll get outraged if it asks for outrage. It's very quick. If someone challenges your beliefs, you're like, I do not like that. I'm going to avoid that. I'm not going to look at that. The availability heuristic means that recent vivid examples seem more probable. And also, if things are repeated again and again and again and again and again, people start to acknowledge in their brain, in their probably lizard level brain, that that might be true. And that psychological sort of tendency makes it extremely hard to battle disinformation when social media is a main source of information for a lot of people. The Dunning-Kruger effect, limited knowledge breeds overconfidence, also known as influencers commenting on health issues. Often wearing lab coats, thank you, saying things your doctor doesn't want you to know and dressing as a doctor at the same time. It's very strange. It's like these things are bizarre, honestly. Social proof, if everyone you know believes something and you belong now to a community where everyone believes something, it's very hard to break out of that. And then motivated reasoning, where is if you've already started to believe something and the alternate to believing it is a very unpleasant thing, like I've been wrong all along and it's a big wrong, it's very hard to budge people from that. They kind of dig in. Intelligence does not immunize against misinformation. So the context in which all of this is happening is a bunch of societal trends. The post-truth era, facts are downplayed in favor of whatever feels right, supports the group that people are in. Populism is rising globally, and this originally just means the population is against the elites. And in this case, the elites are portrayed as experts. So that's been a bit of a shift. So now experts are the elites and the people are, basically rising up against the experts. There's profound distrust of traditional institutions. There's, new media, so the influencers as experts. There's no real verification. Even traditional journalism, I found out when I was doing the journalism course, they actually have very strong standards for how they vet what they publish if they're a true journalist. And there's none of that in social media and influencing, and influencers are often selling you things. And the social media diet, filter bubbles and echo chambers. People are curated into self-reinforcing silos and basically radicalized. So the post-truth era, alternative facts are mainstream. There's no shared basis for truth. There's polarized information ecosystems. Emotional truth, in many cases now, weighs more than factual truth. So you can say things are wrong and still believe them. And people can tell you they're wrong, but they'll still believe them. Expert knowledge is questioned as just another opinion, mostly because at least part of it is people don't understand the nature of hypothesis. That's just a hypothesis. So we're not just fighting false facts, we're fighting the death of facts. This is getting depressing. When everyone's an expert, no one's an expert. And, we do see this in the clinic room because, about 36% of patients in some studies bring conspiracy theories to their appointment. Do your own research means like watching YouTube videos, I think. But that becomes a patient directive. Medical consensus is treated as one perspective, like it's weighed equivalently in the public domain, and feelings validate beliefs. So disinformation taxonomy, common types we encounter: fabricated content, manipulated content, imposter content, false connection, misleading content. And I'm just going to show you an example of that. On the left is the CDC website on autism and vaccines. On the right is a shadow site hosted by the Children's Health Defense, a nonprofit anti-vax organization started by Secretary Kennedy. They look pretty similar. I think they were forced to take it down, but the search engines were taking people to that site. Evidence supporting autism risk. So health disinformation, I think there's some special vulnerabilities associated with that. Health misinformation really is driven by fear and anxiety. It amplifies sharing behavior. Personal anecdotes are very strong in health stories. Distrust of institutions, illusion of knowledge, and frankly, desperation and fear can drive magical thinking. And so when people are scared and sick, logic takes a back seat. And that whole scared piece, I think, really was part of the pandemic. And social media, like a definition, uses mobile and web-based technologies to create highly interactive platforms via which individuals, things shifted here, and communities share, co-create, discuss, and modify user-generated content. I don't know. This thing has rendered all my carefully chosen fonts into garbage, and I apologize for that. It's changed things. So someone should update the PowerPoint on this machine. But the whole point here is that they're highly interactive platforms. Individuals and communities share, they create their own silo, and they create their own content. So now the people are the content. And algorithms are really, really an issue here because they select content based on your social context, personal preferences, and digital trace data. I just was looking at something that suggested that if you start a TikTok account identifying as a teen male, you start getting fed an algorithm of essentially highly misogynistic content within like half a day. That's very frightening. But on a science basis, if you start out with general queries, it doesn't necessarily take you to science. And if you click on something that looks like startling or interesting to you or shocking to you, it will feed you more of that. So algorithms can actually block most credible information from ever reaching audiences that are not starting off engaged in credible science. So my feed is not going to look like a general person's feed because I search different. But there's a lot of highly curated material being fed. And there's challenges with speed, reach, and irreversibility. These are the other quotes. Falsehood flies and truth comes limping after, so that when men come to be undeceived, it's too late. The jest is over and the tale has had its effect. 1710. And things didn't travel as fast then. I think it involved horses and boats. Now with social media, it's different, but a lie can run around the world before the truth has got its boots on. That's from Terry Pratchett. So special case in infectious diseases. I'm going to argue that ID is a special case for misinformation because there's exponential spread for pathogens and misinformation. There's network effects like herd immunity and herd misinformation. There's life or death stakes, and public trust is essential because we need to have trust to have an effective outbreak response. And it really comes down to a politicized notion that infections inherently imply possible risk to others based on your choices. And so that kind of is where the rubber hits the road in a lot of people's thinking about, constraining one's freedoms, right? So, public health, the saying is your right to swing your fist ends where my nose begins. But in reality, some people are not caring where your nose begins. So, there's all these greatest hits of infectious diseases disinformation that a lot of us are familiar with. I'm not going to go into them, but every single myth costs lives. And the virality of conspiracy theories in specific is a bit of a unique area because there's an evolutionary lens here. People evolved to detect threats. Now in our environment, there's not that many tigers behind bushes, but we're still looking for threats. Pollution detection. Invisible dark agents are something that we're also wired to look for. So if you see strangers and the strangers aren't someone you know and they might be sick, you're wary of them. Likewise, people worry about invisible threats like, I don't know, radio waves, like nanoparticles, like viruses and bacteria. Protecting the weak is another evolutionary urge. And that means that conspiracy theories often paint these terrible, terrible things that could happen to innocent and pure victims. That's a common thread. There's the issue of wanting to find your tribe. Who has your back? Who's your group? And once you find your group, you bond with them and you share a worldview. And uncertainty reduction, people just really like a simple explanation. And in science and medicine, we don't got that. So there's all sorts of things that conspiracy theories feed in the psychology, and actually they travel together. If you've accepted one, you're more likely to accept more. So success stories, there are some. I think there needs to be a little more dedicated attention given to this. Counter disinformation examples can start with education. So having good public education is essential. But Finland's education system starts teaching kids critical thinking from elementary school, and those kids are more resistant to misinformation. Taiwan has real-time fact-checking. There's been some good examples of Indigenous community engagement, so cultural attunement in messaging. There's lots of physician influencers who've gained very large followings, and they're using the physician-based trust in a public-facing way. and there's local health champions. And so antidotes do exist. We need to scale it and understand it better. There's a paper here where they were talking about combating misinformation as a core function of public health, and this group put together basically disinformation team in a local public health unit, where they were basically surveying and monitoring for disinformation that was arising and quickly providing useful information to a network of people in the community who were engaged with the community. And they found out a bunch of things. They found out that people prefer information a certain way, that they had to engage with certain partners, and there's lots of things that were learned in this on-the-ground example of real-time disinformation busting for a specific population. And there's actually an interesting BMJ paper that just came out on the social determinants of misinformation. And, you know, misinformation is less intentionally harmful than disinformation, but it's more prevalent. And it's a meta risk, which means that it influences and amplifies all the other major health risks. And so it is a big topic. The key areas to look at are susceptibility to misinformation and equality of access to or ability to identify accurate and credible health-related information. And there's all sorts of work that has to be done in this domain that needs dedicated support. These are some examples from the group I talked about before. And, you know, I actually think that at the end of the day, we have to do research and we have to theorize, but we also have to get boots on the ground, because at the end of the day, it's really also about relationships and trust. I'm going to kind of skip this bit. I'll just say that every consultation can become a battle against Dr. Google, especially if you're trying to get someone to take a vaccine, and that it causes burnout. I mean, if you're in a health-related science medical field and you're just seeing things coming forward day after day that are wrong and harmful, it becomes exhausting, and people basically mentally retreat. They don't want to engage with it anymore. And I think that that's a real risk. And I think that it's one of the reasons why people have been wary of engaging, honestly. And so, you know, one cure in terms of the doctor-patient relationship is trust, empathy, stories stick better than statistics, and trying to maintain your connection, even if someone is holding beliefs that you think are harmful to them. And so there's a call to action here, and it includes a call to action for researchers. There's a lot of barriers to engaging with misinformation. There's time constraints, there's training gaps, there's emotional labor, there's fear of backlash, there's online harassments, there's death threats, there's systemic pressures. But we still need to diagnose misinformation. We need to treat with evidence-based communication. We need to prevent. Pre-bunking or pre-inoculation is getting out ahead of it and saying, you're at risk of getting bad information about this. Here's some good information. And actually calling out the fact that people might be exposed to bad information. There's some systemic solutions that need to be adopted by public health, and there's follow-up. You have to monitor and adapt what's going on. So the question isn't whether to act, the question is how to act. And so I have a couple slides about what to do. So, you know, everyone in the room could do something. It might even just be thinking before you click share if you're on social media. What's your next step? How will you use your trusted voice? And what's your role in the cure? So one thing is every person's visible response matters. So if you see quality information, just make sure you check yourself. Check yourself for confirmation bias. Notice and comment when you see good information. Be vocal about what you won't tolerate. And so I don't always, like when I was more, I kind of am taking a bit of a social media step back for the last several months just because it was too much. But I used to occasionally just pop up in some, like I'd pick one piece of horrible disinformation a day, pop up and say, I don't think that that is good information, and this is why, here's a link. And then leave. I would not get into it any further. I just wanted there to be a signpost in that post that showed someone with relevant credentials was saying, no, that's wrong. And that creates at least some pushback, even though I don't think I'm going to change the mind of the person posting it at all, like not even a chance. But there's a lot of quiet watchers. Make concrete requests. Don't settle for the best of a bad lot. And disaster paralyzes, people retreat, try to feel safe. And so if people were to engage in more misinformation work, pull together because solidarity helps. It's very isolating. So as professionals, there's an opportunity to help build a response to misinformation. And social media is the most obvious place one might think of. I won't go into this a lot, but I will say that there's some pretty compelling data that people who are researchers who have an effective social media presence, even just to other researchers, get higher profile collaborations and ultimately do better in grant funding and get a lot more attention for their papers, even though social media is currently a pit. And that is basically using it like a bulletin board for your own people. If everyone took one step further and made sure that there was a plain language summary, and even if you didn't choose to engage, use the bulletin board to include a plain language summary as to why your work is relevant, that's creating a lot more material to work with for people who are looking to improve science literacy and reduce misinformation problems. So even that basic step of everything you write can have a plain language summary, and that should be put somewhere, is really potentially transformational in terms of just the material that's available. There's a lot of problems with this too, and I have a separate talk on that. So, you know, fact check before sharing, engage respectfully, use your platform thoughtfully. Medium level, write for lay audience, participate in public forums, train in science communication. High level media spokesperson training, I still have to do that. Policy advocacy and research collaboration. And just to bring it back to research, I want to give an example of that last part because it might seem a little bit vague. So I'm a co-investigator. I can't remember which one it is now. There's a guy in philosophy too. New Frontiers in Research Fund Transformation Grant, letter of intent stage, Lara Mahal, next generation mRNA therapeutics, breaking barriers with programmable mRNA technology. So automatically that sounds super cool, right? But in today's environment where states are actually enacting legislation to not allow the use of mRNA, I'm not kidding. Like, I don't know, all life will seize if we take that to its final conclusion. But that is a bit of a hot potato. And so in the grant, there is aim number 4, explore the foundations of trust in science, develop an iterative communication strategy to educate the public and overcome misinformation in the RNA therapeutic space. And there's actually a budget item that I've been kind of trying to work out if it gets past LOI, because I think there's an opportunity to treat this like a quality improvement cycle. So have an engagement platform with actual real people and get feedback and improve it. And I think that that's a nice example because it's part of a grant that is very sciencey, but it's trying to get ahead of potential problems and is trying to contribute to a better understanding of misinformation and disinformation in a science space. So I'm going to leave it there with this image. You can have a whole bunch of different things in the water. And I'm just going to point out that every single piece of clear, good quality information that you put into the stream helps dilute the other stuff. And that's it. I'll take questions.
Fantastic. Thank you so much. I mean, you probably have enough slides to talk for hours on this.
Yeah, I can give several more talks if you want.
Yeah, I'm quite likely. Are there any questions in the room at this time?
Or comments.
Given control governments are trying to exert over public health and the healthcare system and their ability to decision-based evidence making, how do you, I mean, we have to fight that, and I know you've done it, but how do you think we change that narrative?
You know, I think the question is about basically encroachment of elected officials into health and science-based decision-making, which I think is an increasing threat. It's an existential threat, honestly, to both practicing medicine and doing research. It's like massive. And I think the only place where you can effectively battle that is in a ballot box, which is an intermittent opportunity, or in the court of public opinion. Like, honestly, just trying to, and the problem right now, I think, is that people who have concerns are in a bubble and they're talking to each other. And the hard work is getting to people who disagree with what you're saying and trying to find some narrative that you can have a common discussion around. It's always a good idea to start with something you can agree on, by the way, and then work from there. But that is really very labor intensive. But I don't see a way around that. And I actually am very troubled by the fact that I don't see resources for that. because volunteerism can get you so far before you crash and burn hard. I saw a question there or comment or not. Nope.
Here, I'll throw it one then, just briefly, just from, so we have a lot of trainees in the room and a lot of people doing different research projects and programs that way. And just highlighting some of your tips and just even like low level little things that you can do and all of that, which I really appreciate just from all of that, to like doing lay summaries, talking about your research to other groups, putting up a little post here and there. You know what, sometimes those little things, it just takes those little steps. So do like you said, doing what you can to kind of like
pro tip. Almost any of the AI, large language AI models can do a good draft of a plain language summary for you. It saves a lot of time, and you can actually instruct it to be lively and engaging and approachable, and then put in the abstract, and it'll give you a lay summary that you can work with. And let me tell you, that makes people's lives so much easier because it's hard to do that. It's hard to get out of our own jargon. And if you do that, it gives you a starting place for something that, like I said, you might not want to get down and fight with people who don't believe in germs. But you might actually just put something out to inform people about what your lab is up to in a way that the average person, should they come across it, can understand what your lab is up to.
So I guess the question or the comment I was going to ask you about, you know, you and I have had lots of conversations about the benefits and dangers of AI. Any other comments about how AI, particularly the non-optional AI summaries in Google searches now, do you think that those are going to be helpful? Because I can see how they could be helpful, but I also see how they could be dangerous.
I think that so far they look more helpful than not. But what a lot of people don't understand is that if you have an AI search answer, it is a consensus generator. It's not a truth generator. And so basically it's based on the input that it's drawing from. And so if you have a highly biased body of information, that becomes part of its data, it will spit that out for you without any criticism whatsoever. So,
I mean, one of the dangers you pointed out was that the algorithms are currently sorting us into alternate reality. This may be a way to get, I mean, does it get you out of
there's a very, yeah, I mean, I think that there is some value in knowing what the consensus is, but then you also have to look at, you know, how do you actually try to make these systems work for the public good? And actually, the European Union is way ahead of us on a lot of this stuff in terms of trying to figure out what to do about social media having no boundaries except for those that they set for themselves. And there's some early steps towards, you know, basically fact checking, for example, on Facebook that have disappeared now. So I think that there's a lot of threats, but there's also some opportunities in the AI space. And like any tool, it depends on so much on how it's used. And I actually think there probably needs to be a little bit of concerted effort looking at the implications of that, because people can generate disinformation very easily as well. That looks pretty darn compelling. And so basically we're reaching a state where people are willing to trust the results of an AI search. And yet everything that's being presented to them might actually not be true. Every single thing, including the photos. You can't even always count the fingers and know for sure anymore because they're getting better at fingers. So it's a jungle out there.
I am going to turn it over to Dr. Hong, and she's going to introduce our flash talks. So these are very brief 2-minute presentations by our students in the division to highlight again the research that they've done. Dr. Hong has the unenviable task of keeping people on time, which is not easy, but we'll turn it over to her. She's a good taskmaster. So we'll go ahead and get started.
Thank you. Yeah, thanks, Steph. This is my third year doing it. I think it's because I ran a pretty tight ship. So we'll see if we can keep things on time. Each of the talks are 2 minutes, as mentioned. We'll keep moving through each of them. And I believe that all the questions will be saved for the discussion period during lunch. Okay, so the first presenter will be Tanya Swandork. is it perfect? Who with her preceptor, Dr. Severa, and she's presenting homologous composition of plant and human-derived EVs suggest a potential dietary origin of circulating plasma EVs.
Good morning, everyone. My name is Tanya. I work with Dr. Severa. So extracellular vesicles are small lipid bilayer particles secreted by cells. They're believed to play important roles in intercellular communication. They have been extensively studied in humans. However, much less is known about EVs from plants. Our study aims to explore whether plant-derived EVs share structural or molecular similarities with human-derived EVs. We use transmission electron microscope to visualize the structure of starch nanoparticles, which reveal the lipid bilayer membranes and 3 size ranges, consistent with known EV size categories. These structures stain positively with arcadine orange, indicating the presence of cardiolipin, a component typically found in lipid membranes. They also stain positively with Alcium blue and with germagglutinin, supporting the presence of glycosaminoglycans and sialic acid. Through immunofluorescence, we detected key proteoglycans like perlecan, keratan sulfate, and heparan sulfate. Keratan sulfate and specific EV markers were further validated by Western blot. Moreover, when we analyzed the proteins of EVs isolated from the plasma of 4 healthy individuals using plant EVs as positive control, we detected plant-specific chloroplast, mitochondrial, and thylakoid proteins. Thylakoids are membrane-bound compartments within chloroplast and responsible for photosynthesis, and COX-4 is a protein present in both plants and animals. This table summarizes our findings and reveals that plant-derived EVs and human-derived EVs share many similarities. In conclusion, the presence of plant-specific proteins in human EVs suggests that at least a subset of circulating EVs may come from dietary sources. Thank you.
Hey, thanks very much, Tanya. Next, we have Brian Evans, preceptors Dr. Smith, presenting on measures to decrease catheter-related bloodstream infections in the ICU.
Okay. Perfect. Yeah, thanks so much. So yeah, I'm Brian Evans. I'm one of the final year subspecialty residents finishing up my time here in Edmonton. So just an update of what I was working on this year. So I worked as somewhat straightforward, but an interesting project with Dr. Smith as my supervisor, trying to create a useful educational tool and resource to help educate and inform residents rotating through the ICUs. So this is piggybacked on other new documents and resources, which include a recent guideline checklist and webinar that was developed by the Critical Care Performance Improvement and Integration Network here, led by Dr. Schlegel, in tandem with Infection Prevention and Control. The overall objective of my project was to create a multimedia presentation which can efficiently review the best practices and available resources for rotating residents through our academic ICUs and to provide evidence and context surrounding those recommendations. Further context behind this initiatives is that CLABSI rates actually increased across Alberta's intensive care units over the COVID-19 pandemic, which you can sort of see in that middle table there, and have subsequently remained elevated. A 2023 environmental scan completed with clinicians across Alberta identified opportunities for provincial standardization of best practices related to education, insertion, and ongoing maintenance of central lines. The educational session was developed with a focus on the primary literature, as well as guideline recommendations with some case-based teaching, with an emphasis on the recently updated local guidelines for best practices for rotating residents in the ICU. This session covers various points of intervention and the primary literature and recent guidelines in relation to different points, such as choice of antiseptic, chlorhexidine impregnated dressings, site selection for line insertion, drying time, and the use of checklists during insertion and maintenance. And that's it. Thank you.
Thank you, Brian. Next up, we have Carmen C. Up again, her preceptor was Dr. Connor O'Neill and Molly Lynn, and her talk is on C. difficile and rotavirus mixed pathogen outbreak in an acute care unit in Edmonton, Alberta from March to April of 2025.
Thank you. So rotavirus is a commonly recognized cause of gastroenteritis in infants and young children. However, it is underappreciated in adults. There's currently no treatment or post-exposure prophylaxis, and therefore infection prevention and control, or IPC measures, remain the most important intervention. We report on an outbreak investigation that was conducted on a geriatric acute care unit at the Misericordia Hospital. As seen in the EPI curve between March 20th and April 10th, 5 C. diff and 6 rotavirus cases were identified on a single acute care unit. One patient was co-infected with both pathogens. 3 C. diff cases were epi-linked to the same patient room, and the median age of those affected was 87. The outbreak was closed approximately 3 weeks following the outbreak measures that were implemented. As per the timeline seen in the detailed outbreak table, IPC measures were implemented in accordance with Covenant IPC guidelines. An initial hand hygiene audit of 21 opportunities revealed only 66.7% compliance with follow-up education provided, and then a subsequent hand hygiene audit of 110 opportunities showed an increased compliance to 93.6%. Fluorescent audits with GlowGerm also revealed improper cleaning of the patient environment and shared equipment, and the ward received targeted cleaning with sporicidal cleans as well. Other areas of concern identified included having staff reporting a difficult time hand washing due to the cold water temperature, staff using the incorrect disinfectants, and staff having their water bottles in the patient nourishment center. To our knowledge, this is the first documented mixed C. diff and rotavirus outbreak in the Canadian acute care setting in the literature, and this underscores the importance of not only considering multiple pathogens in outbreak investigations, but also ensuring that surveillance and diagnostic practices are sufficiently broad. Thank you.
You guys are making my job easy. Everyone is perfectly on time today. Okay, so our next speaker is Emily Kingdon. Her preceptor is Dr. Joel Dax, and her presentation is on pinpointing loss of the cellular component ARF6 in the lineage leading to Giardia.
All right, hi. So Giardia is responsible for over 250 million cases of diarrheal disease every year. It also impacts populations where water supply is an issue, such as the Canadian North or Indigenous populations. Giardia has a highly modified endomembrane system and lacks a lot of canonical compartments, such as the Golgi, endosomes, intermediate vesicle carriers, and also employs mitosomes instead of canonical mitochondria. So since we're talking about the membrane trafficking system, just to orient us a bit in case, but the briefest of introductions. So ARF gaps are going to, small GTPases are going to initiate recruitment of the coat proteins, after which point the ARF gaps are going to impact transport. After budding incision, the vesicles are going to move towards the target membrane, where they will be recognized by tethering proteins and attached via snares. The proteins that we're specifically looking at today are the ARFs, specifically ARF1 and ARF6. So ARF1 in modern cells is found around the ER and the Golgi there, and functions with trafficking, vesicle formation, and coat protein recruitment primarily. ARF6 is often found where the purple star is there by the endosomes, and primarily regulates endosomal and plasma membrane protein function. It's important for us to understand the system, as the vesicle secretion pathway is very important to Giardia's pathogenicity. So previous studies had shown a very reduced version of this system within the metamonads. However, since this publication came out, there have been some new genomes released, specifically expansions within the Basque clade. What we did is we took these new genomes and we incorporated it into our study. Turning your attention to the phylogenetic tree here, the well-supported clade in purple is the ARF6s, and in pink is the ARF1s. And these Basque genomes that I mentioned are sitting within the ARF6s. We took this and did some further analysis to confirm, and did confirm the presence of ARF6 within these Basque genomes, which has not previously been identified. This provides the foundational context for the study, as I will go study in our lab, our partner lab in Switzerland, where they're looking at these proteins functionally. Thank you.
Okay, our next speaker is Felix Runga, preceptor Dr. Doucette, and his research is on the incidence of CMV disease in CMV donor negative, recipient positive, solid organ recipients in Alberta.
Thank you. So it'll be presenting part of our ongoing work. CMV is the most common infection after transplant, and without prophylaxis, especially in the highest risk groups, up to 75% will develop disease. Now, there have been strategies that have been devised for most with high risk, who are the donor positive, recipient negative SOT patients. But in this group where we have the purportedly low risk groups, D minus R plus patients, there are no clear guidelines on what the best strategy is for them. Risk estimates for CMV disease for this group range from 0 to 5%. We do not know what that is for Alberta, and currently we do not have a strategy for following them up. So our aim is to determine the incidence and risk factors for CMV disease in the D minus R plus patient population in the first year post transplant. It's a retrospective study where we are taking heart, liver, and kidney solid organ transplant recipients. We are excluding lung because by definition they get antiviral prophylaxis whenever they are R+, and anybody getting lymphocyte depleting agents. So far we've managed to review 63 solid organ transplant recipients, 38 in liver, 21 in kidney, and 4 in heart. We've found CMV disease in 6%, that's 4 patients, 3 in liver and 1 in kidney. Of these, 1 had colitis and the other 3 had CMV syndrome. There was 1 death. There were also 2 who had CMV infection without really meeting the definition of disease in liver transplants who required therapy. In conclusion, CMV disease may not be an infrequent occurrence in this low-risk population, and on getting the complete data set, it may inform change in strategy as to how we follow up this group. Thank you.
Hey, thank you, Felix. Our next speaker will be Jessica Skorejko. Preceptor would be Dr. Meyer Stevenson, and her research is on the impact of GQ binding ligands on hepatitis B viruses life cycle in vitro.
All right, hi everybody. As many of you know within this room, hepatitis B is one of the main causes of hepatocellular carcinoma or liver failure. This is due to the establishment of the cccDNA. It is a highly stable viral genome that actually mimics the host genome, making targeting and eliminating a challenge. This is where my research comes in. It was recently discovered that within the pre-core promoter region of the cccDNA, which is a vital promoter for the viral progeny, this highly conserved segment of the genome, is enriched in guanosines, which take on an unconventional secondary structure to aid in gene regulation and replication. It was found that if there is a disruption to this G quadruplex, you actually see a reduction in virus in vitro. With that being said, we are taking G quadruplex binding ligands from the cancer fields and adapting it and applying it in HBV. To do this, I use a variety of methods, but primarily it's a transfection model and a reporter assay. Based off of these findings, we found that the ligands will influence the promoter and actually aid in the amount of activity that we see, which suggests that they do bind in some capacity. And then in the transfection assay, you can see in the immunofluorescence staining, this was a proof of concept that as the transfection goes on, there is increased green staining of the core protein or the red arrows because of the images here. Finally, we do look at the nucleic acid level, and we see that with these ligands, TMP-YP4 seems to be the most beneficial at the DNA level upon day 2. However, further in the infection or transfection, all of the drugs seem to have the same susceptibility to the virus. And then finally, at the RNA level, they all seem to have the same impact. With that being said, this data suggests that further looking into how these drugs bind to, and specifically where they bind to the pre-core genome, could aid in a cccDNA targeting therapeutic in the future. Thank you.
Jessica. Right, thank you, Jessica. Our next speaker is Justine Beghin, preceptored by Dr. Meyer Stevenson. Her research is on designing therapeutics against an evolving target, a strategy for probing new proteins of emerging viruses.
Hi, everyone. My name is Justine, and I'll be talking to you today about a platform to screen for potential therapeutic targets against newly emerging viruses or viruses with quick evolutionary turnaround. Influenza is a good model because of its strain diversity. Most viruses, even diverse ones like flu, have a protein that is so essential, it rarely changes, and is very conserved across strains. This makes it a good therapeutic target. For flu, this is its polymerase, or flu pull as I'll call it. Flu pull is essential for viral replication and transcription and is composed of 3 subunits. So our question is, can we design a platform using an aptamer-based approach to find targets that bind and interfere with the functioning of flu pull? My project aims to answer this question using aptamers and a technique called SELEX, or systematic evolution of ligands by exponential enrichment. Aptamers are short DNA or RNA sequences that fold in a specific way to fit potential binding sites on complex protein structures and are alternatives to antibodies. SELEX takes a functional purified protein of interest, in this case flu pull, as you can see here, and cycles through a process of mixing protein with aptamers, binding, washing away non-binders, and then amplifying the stronger binders via PCR. An initial aptamer pool of 40 nucleotide long randomly incorporated single-stranded DNA with primer ends we can recognize in PCR was used in the first round of SELEX. Then the end pool of binders after PCR was cycled through again and again for 8 to 12 rounds to create a final aptamer pool specific to flu pull. Sequencing was done to determine top hits in the aptamer pool, such as those highlighted in green in this chart. These hits were mixed with flu pull and functionality assays to determine if flu pull function was negatively impacted by aptamer binding. If we saw this peak on the left there decrease, that would indicate an aptamer could be a good base for designing a new therapeutic against flu pull and should be further investigated. So to conclude, this platform is highly versatile and can be modified for universality across different strains of flu pull or the essential conserved proteins of other emerging viruses.
Okay, our next speaker is Kira Spidersky. Yeah. And her preceptor is Dr. Meyer Stevenson, and her talk will be on G4 quadraduplex in the HBV pre-core promoter, defining structure and function using mutational, computational, and proteomic studies.
Okay, hi everyone. So as Jessica has mentioned, one of our key research areas is HAP-B, and in particular, the same region over here found in the pre-core promoter that is vital for the viral RNA expression. So as she mentioned, it's able to fold in a non-conventional secondary DNA structure called the G quadruplex. So to facilitate therapeutic development, we decided to perform several studies, including promoter mutagenesis, computational predictions, and proteomic analysis to determine function and the structure of the region. So starting with promoter mutagenesis, we've shown that mutations in the early nucleotides here are more integral to the structure of the quadruplex, while latter might exist in several conformations naturally. We also showed that the HBV quadruplex can be substituted for a human quadruplex in the context of the viral promoter to a varying degree here, as well as that a drug supposedly binding at the site is able to prevent transcriptional activation from the virus. We've then moved on to computational predictions, where we elucidated several candidates for the confirmation, and we are currently testing them in MD simulations. As well, we've performed proteomic analysis, where we compared our oligomer representing the quadruplex against its double-stranded counterpart. So it has the sequence, but it does not have the right shape or the randomized 33-mer sequence. So we first confirmed that the quadruplex is able to find in its fold in its proper shape, as well that it can be separated and it's a homogeneous species. We've then transfected them into cell culture and showed that they can A, be tracked to the nucleus, B, can compete with other quadruplexes and binding sites for protein binding. Our initial proteomic analysis yielded that quadruplexes are able to bind common transcription factors as well as quadruplex-specific proteins that all could be relevant for hepatitis B infection.
Thank you, Kira. Our next speaker is Roy Hutching. Oh, okay. Our next presenter is Roy Hutchings, and he is presenting virtually today.
Can you guys
His
hear me?
His preceptor is Dr. Chen, and his talk is on examination of, yeah, okay, that's all there is. Examination of echinocandin appropriateness in hospitalized adult patients at a Canadian tertiary care center.
Can you guys hear me okay?
We can hear you, Roy. We're just trying to get your slide presented. So I will let you know once we have it on the screen, and then you can start.
Okay, sounds good.
Thank you.
Yeah. Okay, your slide is now presented. Go ahead, Roy.
Sounds good. Hi, everyone. My name's Roy. Echinocandins are broad-spectrum antifungal drugs. They're established antimicrobial stewardship targets owing to their toxicity and cost and risk of inducing the development of antifungal resistance. And at the University of Alberta Hospital, the usage of echinocandins has been increasing, but we don't know whether that usage is appropriate. So we piloted the addition of echinocandins to our prospective audit and feedback program with a primary objective of evaluating the appropriateness. We conducted a prospective audit and feedback study from June 1st to 30th during my ASP rotation. All new echinocandin prescriptions in adult inpatients at the university were included and audited from Monday to Friday. We assessed the prescriptions as appropriate or not based on local prescribing guidelines and expert opinion. And if assessed as inappropriate, we provided real-time verbal and written feedback to the physicians to optimize the prescription. In total, there were 22 mycofungin prescriptions over the study period. The spread of the indications are shown in the big gray table there. 5 prescriptions were for prophylaxis against Candida empyema in lung transplant recipients, which was the most common indication. And in total, 8 prescriptions were for prophylaxis using a local protocol. Now, for a primary outcome of the 22 prescriptions, 5 were assessed as inappropriate and needing change. That's 23%. If we exclude those given for prophylaxis following local protocol, that goes up to 31%. We provided recommendations in 6 prescriptions, and the acceptance rate was 83%. So auditing echinocandin prescriptions frequently resulted in recommendations and were broadly accepted, although there was a fairly low volume over the study period. So while there may be an opportunity for stewardship optimization of echinocandin prescriptions at our center, we need a larger study to better understand baseline prescribing practices. Thanks.
Thanks very much, Roy. Our next speaker is Verlika Tyagi, and along with her preceptor, Dr. Kabani, and she'll be presenting norovirus and sapovirus associated chronic diarrhea and solid organ transplant. Does viral load correlate with severity of symptoms?
Thank you. Okay, hello everybody. So norovirus and sapovirus are common causes of viral gastroenteritis. In immunosuppressed SOT, these infections can become chronic and difficult to manage. So in our study, we aim to evaluate the relationship between symptom severity and viral load, and also assess the virologic and clinical outcomes associated with different treatments. So this is a prospective study enrolling adult SOT recipients with confirmed norovirus or sapovirus. Participants provide regular stool samples and symptom questionnaires, and patients are being followed for up to 2 years. So far, we have enrolled 8 participants that have provided at least 1 stool sample, and the majority of our patients are infected with norovirus genotype 2. Notably, the only participant with norovirus that did not develop chronic symptoms was a liver transplant recipient on a single immunosuppressive drug. The figure below shows the viral load over time in 6 patients with norovirus. What's interesting is for the participant with the multiple samples in blue, their symptoms did not resolve until immunosuppressives were reduced, but this decrease in symptom severity did not correlate with the decrease in the viral load. In terms of symptoms and complications, most participants develop chronic diarrhea and vomiting, with the majority requiring hospitalization and IV hydration. In general, sapovirus infections had a shorter duration of symptoms, and these patients cleared the virus and had negative test results. In contrast, norovirus viral load remained detectable in stool samples even after symptom resolution. So in conclusion, norovirus is associated with prolonged symptoms and extended viral shedding in SOT recipients. Limitations include a small sample size and short-term follow-up. Future directions include testing initial clinical samples of our participants and continued enrollment and follow-up.
Okay, thank you. Our final speaker is Cecilia Shi with her preceptor, Dr. Natalie Marshall, and her talk is on unmasking rare Ignatius bacteremia. Clinical history informs modern diagnostics.
Hello, everyone. Today I'm presenting a case where clinical history became the diagnostic compass in mystery gram-negative virus identification and give us the answer days faster than 16S sequencing. So this gram-negative virus were initially worked up in a base lab and the automated identification system failed multiple times. So it was sent to prop lab for 16S sequencing. Together with the isolates, we also got detailed clinical history from ID colleagues. It is a 55-year-old female admitted with polymicrobial bacteremia, secondary to diabetic foot infection, and with critical flag, Mager's infested wounds. So while awaiting 16S, the Brooker Marty at Pop Lab gave us a high scoring but unusual identification, Ignatius larvae. It is unvalidated ID, so the micro team was consulted. Here's a magic moment. The micro training immediately recalled this association between this organism and the Mager's infestation. And there's also a new session of Ignatius section added to the latest manual of clinical microbiology. So we're able to compare the patient's isolates to the expected results in the reference material. And everything matches perfectly, including the morphology, the biochemicals, most importantly, the clinical picture. So the micro team was feel comfortable to deliver this preliminary ID with susceptibility guidance. 2 days later, 16S confirmed this Ignatius species. And the patient have a successful infection resolution with proper antibiotics and BK eventually. So about Ignatius it's a commercial in fly larvae. Handful of human cases reported, can cause bacteremia and severe sepsis. No breakpoints for antimicrobial, but overall susceptible to beta-lactam. So what I learned from this case is that the modern diagnostic sometimes can lack validation for these rare organisms. So clinical history combined with updated micro references would be very helpful and sometimes so transformative to give us the confidence to act on quickly on this unusual idea and deliver timely patient care. Thank you.
Thank you, Cecilia. So this concludes our round of flash talks. Can we all just give a round of applause to our 11 fantastic trainees who shared their research with us today?
All right. Thanks very much, everyone. So it is now my pleasure to announce the speaker for the Dr. Jeff Taylor Lectureship. This lectureship was established in Dr. Taylor's name 4 years ago after his untimely death in 2020. Dr. Taylor had many areas of interest within infectious diseases, and infection prevention and control was one of them. So Dr. Susan Huang is this year's Jeff Taylor Lectureship speaker. She is a Chancellor's Professor of in the Department of Medicine, Division of Infectious Diseases at the University of California, Irvine School of Medicine. And she's the Medical Director of Epidemiology and Infection Prevention at UCI Health. For the past 20 years, she has been studying healthcare-associated infections with a focus on multi-drug resistant organisms. And her epidemiologic research seeks to identify the burden, risk factors, and preventative strategies for containment. And she has conducted several large randomized trials demonstrating the value of decolonization on disease prevention. Dr. Huang has over 200 peer-reviewed publications and has received 2 top U.S. Clinical Research Achievement Awards from the Clinical Research Forum. She is a voting member of the U.S. Presidential Advisory Council on Combating Antibiotic-Resistant Bacteria and has served as a past member of the U.S. Healthcare Infection Control Practices Advisory Committee and the IDSA Antibiotic Resistance Committee. So please welcome Dr. Susan Huang.
It's a great pleasure to be here. Thank you for that kind invitation. I thought you'll notice that the talk is a little different from the title is a little bit different. I thought, given that this is Research Day, that I might combine a bit about pragmatic trials. What does that mean? And what are the practical experiences from that with the scientific concept of decolonization to prevent MDROs and infections. There we go. All right. Thank you. So I am conducting studies and trials in which contributed antiseptic product is being given to nursing homes and hospitalized patients. So I thought I would start with the published story. You can read more about it because it's published, and then go into a number of things that are unpublished and the things that kind of the backstory behind a lot of the things that you do. So we'll talk a bit about the science, and then we'll talk a bit about the conduct, and that might help those of you who may not have a particular interest in decolonization, but might have an interest in the way in which trials are conducted. So for the science, healthcare-associated infections, as you all know, are a pretty major problem across the world. In 2000, it became a pretty fiery topic in the United States and elsewhere. And basically, they were recognizing that even in the best of circumstances, in our best hospitals, we were causing what they said is the equivalent of a 747 jetliner crashing every other day with no survivors. And those were the amounts of preventable mistakes that were happening in US hospitals at that time. So they devised a number of things, including what they considered to be a 5-year action plan. It started in 2009. It's been going on ever since. And the first time this came up, there were numbers that were attributed to this that we were all supposed to achieve. These numbers crossed a number of infections, things that were device associated, like central line associated bloodstream infections, or things related to catheters for urinary, for urine. Procedural associated things like surgical site infections, and then, of course, the litany of multi-drug resistant organisms. And you can see that the 5-year targets there were pretty large. And at the time, we had no idea how we were actually going to achieve this. And this led us to think a lot about whether or not you have to target everything one by one, or if there's something that could actually span a couple of infections and give you a couple of wins all at once. This also is pretty true for multi-drug resistant organisms. So we have only increased our list. None of them go away. They start to come up every couple of years. A new one shows up. The most recent one is Candida auris, with its new name now, Candidozyma auris. And effectively, if you look at this list, about 10 or 15% of people in the hospital, if you screen heavily, are positive for a multi-drug resistant organisms. A good number of them we're not aware of. If you look at long-term care, it's 65% or higher. And if you have ventilator-associated long-term care hospitals, those it's 80% or higher. And so these are pretty strong reservoirs for these organisms. And again, long time ago, when MRSA first came into the horizon, we wanted to target one by each, swab different body sites, do different things to target their eradication. And now that we have so many on the list, it raises again the question about whether or not we can do horizontal approaches, this idea that you could have a wide swath of things that might be made better, in addition to having targeted approaches. So I'm going to talk about decolonization today with the definition that we're talking about things that are topical and not things that are systemic. So things that actually are placed onto the body, generally antiseptic soaps and nasal ointments, which could be either antimicrobial or it could be an antiseptic, to reduce the body's bacteria when we become our own worst enemies. So when does that happen? It happens when our skin integrity is no longer intact, when we have devices or we can't clear secretion. So a lot of these things that we're talking about happen within the context of hospitalizations or nursing home or a post-discharge time when those things have been delivered, like a surgical incision, for example. During those moments, it's been shown over and over again, if you look in the microbial distribution of infections, these are things that we carry on our body. They're either human commensals or things that can colonize us, and then we get into trouble. So I'm going to talk about the most common regimens that have been studied in large-scale trials, and that's going to be chlorhexidine, which is an antiseptic soap that's been around for a very, very long time, or nasal mupirocin, an antibiotic, or povidone iodine. So this is, and actually one thing I'll say about chlorhexidine, just briefly. Chlorhexidine is an antiseptic that was discovered over 65 years ago. So all those things that are there have been kind of well vetted for their safety structure. Chlorhexidine is over the counter. You can generally find it in a number of pharmacies. And why would it work better than soap and water? So soap and water or alcohol hand rub, you clean your hands, your hands are clean, and then you touch something and you're immediately recontaminated. Chlorhexidine actually binds to the proteins of your skin and stays there for 24 hours if you apply it correctly. And during that time, while it's staying there, it actually continues to provide cytal kill for the pathogens I mentioned, the MDROs that I mentioned. And so ideally, in the best case scenario, you can actually protect a patient throughout their hospital stay by doing daily chlorhexidine bathing, or even in long-term care where they don't do daily chlorhexidine, you can actually use it periodically. They bathe maybe 2 or 3 times a week, and you still get that benefit because it takes time to be recolonized. So that's the concept of why this may be better than routine soaps and water. So humans, of course, we are well known to have this cascade of unfortunate events in which we share pathogens with one another. So we are a massive plume of bacteria, and this has been shown with settle plates and other things, but you can imagine that we're just a cloud of bacteria that we walk around with. And as we walk around, we deposit our bacteria on environmental surfaces, and our bacteria are very, very hardy, and they're perfectly content to sit on inanimate objects for days, weeks, even months. If we don't clean it, which we're not particularly good at doing, you can pick it up, and then you can bring it with you as a provider, room to room to room, patient to patient to patient. And we know in a series of studies that have been done that the greatest risk factor for developing an infection with a multi-drug resistant organism is becoming a carrier of a multi-drug resistant organism. So colonization is something that we should worry about and want to prevent. We also know that there's a number of things that we do and need to do in infection prevention, which you can see listed there. But honestly, if you look at the things that we do for people with multi-drug resistant organisms, it's really important to start at the top of the cascade. So one of the reasons why decolonization is so attractive is it actually starts at the outset, when things are like a trickling stream before it becomes a raging river as it goes down through all these different things that can happen as pathogens spread. And so preventing shedding has been shown, I don't have the time to go through it, but in a number of articles, it's been shown that if you stop the shedding, you will stop environmental contamination, not stop, you'll reduce environmental contamination, you'll reduce the spread to other people, you'll reduce the amount of contaminations on nurses' hands, and you'll ultimately be able to reduce incidence and infection. The other reason why I really like decolonization, though, is because if you think about all the things that we do in infection prevention, clean our hands, clean the environment, wear gowns and gloves, it's all intended to stop pathogens from going from one person who already has something to someone who does not. It does nothing for the person who already has it. And I told you that people already have it. They already have an MDRO, 10 or 15% in a hospital, 65% in long-term care, upwards of 80% in ventilator-associated settings. So decolonization is the only infection prevention strategy that we have that does something for the person who already has it. Otherwise, what you're saying is, too bad for you. I'm only going to protect the people around you who don't yet have what you have. So chlorhexidine, as I mentioned, was developed in the UK years and years ago. It's at least 65 years old. It was used initially for hand antisepsis, and then it went into the dental hygiene world. It's still used heavily for periodontitis. It was then used by Dennis Mackey to show definitively that chlorhexidine is the better thing to clean the skin with, better than alcohol, better than betadine when you place a central line. They've also gone to show that chlorhexidine alcohol is better than betadine alcohol when you're placing central lines as well. Then the world moved into surgery, and they demonstrated that, again, definitively chlorhexidine is the better agent when you're comparing it against betadine and alcohol to clean for surgical incisions. we moved into surgical bathing, and there's a whole group of literature that was looking at how we can prevent people from having Staph aureus infections after surgery, because Staph aureus is a commensal. A third of us are carriers at any given day. And ultimately, they did a lot of screening to find people who are positive and then decolonize them right before surgery. Then we came into the 2010s, and I'm going to start our story there. This idea that as we were looking at the world, we were looking at all of these infections that we were supposed to prevent. We were looking at all these multi-drug resistant organisms that just kept accruing. And ultimately, we wanted to do something to target people who are at greatest risk. So populations, not necessarily one by each persons. And so I'm going to start by talking about some pragmatic cluster randomized trials. At around the same time in the United States, it became really interesting. The people became interested in what they considered to be pragmatic trials. They were tired of looking at perfect trials. That is, efficacy trials are generally designed so that you get the perfect person, you highly curate who enters the trial, and ultimately you have a perfect understanding of what a drug or an intervention will do. But then when you roll it out in real life, you don't see that effect. And so that's why, for example, with pneumococcal vaccine, we have a 65% effectiveness when the trials would show a 90% target efficacy. So people started to say, you know what, it's really, really hard. We spend all this money, we look at this thing, we know what the drug does, but when we roll it out, we don't know what to expect. We don't know if we're going to get 90% or we're going to get 60%. Why don't we do pragmatic trials? Why don't we do trials that enroll as broadly a swath of people as possible and try to understand what happens under routine care? So pragmatic cluster randomized trials means that you randomize not a person, but a group. And so these are types of things that are best tailored towards a hospital or a clinic or a unit. And generally that means we're talking about things that are done for quality improvement or protocols or things that are normally done under routine standard of care and comparing one standard against another. So pragmatic cluster randomized trials are generally for the evaluation of QI strategies. And now this is going to be considered QI research. So now merging, some people say I do QI, some people say I do research. This is QI research where you take QI strategies, you pit them head to head in a really, really robust way to understand which one is in fact better than the other. QI strategies have to be studied in the way that they are applied. So it doesn't make sense to parachute a whole bunch of research staff in. You want to understand what happens when routine people, your routine nurses, your routine people who operationally do what runs your hospital, when they change their particular practice from one intervention to the next. That also means that these are generally minimal risk. And generally you have full waiver of informed consent, not verbal consent, but an absolute waiver of informed consent. When might that be? That might be when you're studying, for example, antibiotic stewardship strategies, behavioral strategies, something where you're looking at, in this case, I'll talk about bathing. You know, the patients don't get to pick their soap. They don't get to come in and say, I want this soap versus this soap. This is standard of care. So if you're going to pick one soap or the other, you get to do that on the hospital level or on the unit level. It also allows you to study things that are highly clustered. So as I mentioned, physician prescribing is clustered. We tend to do what our peers do around us in a group. We tend to be taught together. You have a cohort that goes through fellowship. You are more similar to your peers that you train with than you are to other places. So there is a style and a trajectory within an institution that people tend to be more similar together than you are between hospitals. And for that reason, clustering needs to be accounted for. So there's some specific statistical ways in which you can do that and need to do that. So I'm now going to kind of go through the science of cluster randomized trials that have looked at decolonization, and then we can talk about kind of the background between them. In 2013, 3 major trials came out. The first one was by Mike Climo, published in the New England Journal, looking at academic adult ICUs, about 8,000 patients, demonstrating that chlorhexidine, if you use it as routine bathing for everybody in an ICU, you can reduce the spread of multi-drug resistant organisms, particularly VRE more than MRSA in his particular study, and also reduce all-cause bloodstream infections. So not central line associated infections, but in academic adult ICUs, many, many people do have lines. Then in the same year, Erin Millstone published a pediatric study. Now, pediatric studies are very, very difficult to do, and they do not get to waive informed consent in the United States. And so this is a full consented trial. That means that they could really only do an as treated evaluation rather than an as randomized. 10 pediatric academic ICUs, 1,500 patients. And they also showed they could reduce all-cause bloodstream infections by 36%. So then we did the reduced MRSA trial. This is a large-scale trial done now in community hospitals. So academic hospitals are a very small fraction, usually about 9–10% of US hospitals. The vast majority of them are community hospitals. They can be complicated. They can be tertiary care, but they are generally not teaching hospitals. So 43 community hospitals in a health system known as HCA, previously known as Hospital Corporation of America. This is a for-profit healthcare system. They have about 200 hospitals across the United States. So this is a 43 hospital trial, 74 adult ICUs, 74,000 patients in this particular trial. And we were interested in understanding whether or not you could just screen and isolate. So you swab the nose of everybody who comes into the ICU, and if they have a positive Nereus culture, or if you happen to know they have MRSA from any other reason, you isolate them. That is gowns, gloves, and generally ICUs are single rooms. Or you do targeted decolonization. Same thing, you screen, you isolate, but now if they're positive, they get 5 days of chlorhexidine, 5 days of mupirocin. Or you say, wow, everybody in here is critically ill. We know that a third of humans have Staph aureus. We know that the swab is 65% good at any one day for finding people who are Staph aureus carriers. We are going to stop screening and save all that money, and we're going to give everybody chlorhexidine bathing, and we're going to give everybody 5 days of mupirocin. And so that is the way that this trial was set up, and it found that universal decolonization was by far and away the most effective arm. It reduced MRSA clinical cultures by 37% and reduced all-cause bloodstream infections by 44%. And this is after a decade in the United States of bringing a lot of our device-associated bloodstream infections way, way down. So to see a 44% effect on top of that was actually surprising even to us, the investigators. The impact in a pragmatic trial, because again, we did not parachute researchers to do the bathing. We did a lot of training, we did a lot of cheering. So this is on about 80% adherence. So as you can imagine, people have reasons why they miss a dose. They're in radiology, all sorts of reasons they refuse or they're allergic. We followed that trial with another study in the ICUs, and this was looking at whether or not you really have to give everybody mupirocin. There's a lot of questions about mupirocin being an antibiotic. Do you really want to give it to everybody in an ICU? Are you going to breed resistance? Resistance we know already exists to mupirocin in different parts of the United States. And so we did what was effectively a non-inferiority trial, and that means that you have to have high numbers to meet your non-inferiority margins. So a non-inferiority trial comparing chlorhexidine bathing with mupirocin head-to-head against chlorhexidine bathing with povidone iodine or iodophor in the nose. And so this was the mupirocin iodophor swap-out trial, 137 hospitals, 233 adult ICUs, 353,000 patients in this trial. And this was, again, a two-group pragmatic cluster randomized trial. And it demonstrated that mupirocin was better. It just was better, 18% better in reducing Staph aureus. And in this system, the HCA system, we had done the reduced MRSA trial about 10 years previously. As soon as that trial came out, they adopted universal decolonization in every ICU in their entire system. So they had been giving mupirocin to every adult ICU patient in their system for nearly 10 years by the time the mupirocin iodophor swap-out trial occurred. And that gave us the opportunity to determine whether or not resistance had caused a loss of clinical benefit. So when you do those two trials head-to-head and you compare the arm that's doing the decolonization, you can see that there is, I don't have all the slides, but it's published. There's no decrement in the clinical benefit after 10 years of continuous use. So very, very reassuring about this particular extremely high-risk population. Again, they're all critically ill. They all have lines, devices. A lot of them have incisions, lots of things to actually protect. So we then went back and said, okay, this is great. We're doing ICU decolonization, and that is the standard of care now in the United States. The last CDC survey showed upwards of 70-something% of hospitals across the United States are using chlorhexidine bathing in their ICUs. And so it was a very natural question about whether or not, if we can protect those that are the most ill in the ICUs, can we do something for them when they're out of the ICUs, the rest of the hospital, the non-ICU group. And it turns out that by the time we were doing this trial, which is known as the ABATE infection trial, people had already made a leap. So they took a leap of faith. They said, you know what, we're doing this in our ICUs. We're doing it. We're trying to get CLABSIs down, central line associated bloodstream infections. We're just going to give it to all these people with lines who are outside of our ICU. So there was a big tendency, even when the trial came out, as things happened, to actually be in advance of the science to try to do a couple of things to reduce the financial risk of having high CLABSI rates, because in the United States, you get penalized. So a good amount of your insurance funds can get withheld if you're in the highest tier bracket. So this was the ABATE infection trial. This was 53 community hospitals, 194 non-ICU units, 340,000 patients. And what we did is we randomized the non-ICU units, hospital by hospital, to either routine care, bathe however you do it with the usual soap that you use, or we're going to swap out chlorhexidine for every place you have soap. So in the shower, it's going to be 4% rinse off chlorhexidine, and in the bed bath, it's going to be 2% leave on chlorhexidine. And if you happen to know, because they were not doing screening in this particular trial, if they happen to know that the person has MRSA, they'll go ahead and add mupirocin as well. So it was targeted mupirocin universal bathing. And what we found in this trial is that decolonization, when you look at the overall group of community hospital patients, there was about an 8% non-significant reduction in multi-drug resistant organisms as well as bloodstream infections, non-significant. And so we went ahead and looked at the medical device group, the group that people were already jumping the gun and actually trying to treat people with medical devices around the country. And we looked at that group, and in that group, we found that it was absolutely true that you could reduce MDROs by 37% and reduce bloodstream infections by 32%. Those people who had lines and devices in this healthcare system were about 10% of the population, and they were responsible for 60% of the bloodstream infection. So a very high yield targeted group to try to prevent the adverse events that happen from having a central line or being a person who needs a central line who then gets into trouble. Adherence in this group, again, very, very typical, around 80%, as most things are by the time you load them out into the real world. So we then thought we should go outside of the hospital. We know that in the United States, there's an incredible pressure to have very, very short lengths of stay, and that's because you get paid a lot of times in a bundled approach. So you get one payment for a certain type of hospitalization. And so the more you can churn and turn those people out and get them to the nursing home or get them home with home health, the more money you can make. So the lengths of stay tend to be pretty short. Lots of average lengths of stay are around 4 or 5 days. Median length of stay definitely in the 3-4 range across community hospitals in our country. So we decided, well, by the time a doctor even knows that you have MRSA, you're gone. So what if we look beyond the hospital stay and try to do something there? If you look at MRSA in the United States, there's been an incredible amount of fatigue about multi-drug resistant organisms. And there is a tendency to say the more common it is, the more I want to forget about it. The more common it is, the more I'm just going to take a laissez-faire approach and just say, you know what, I can go after the rare, but I can't mentally handle all this gowning and gloving and everything for something that is common. And yet, when we look at MRSA, I'll only make some comments about MRSA. It is a human commensal, and it is an incredibly versatile pathogen. It can cause invasive disease in every part of your body. And effectively, that particular pathogen, there was a really nice study that was done, 2021, I think it was published in JAMA, and it looked across the world and looked at continent by continent. So North America is kind of grouped together, and it looks at a 1-day stay in the ICU, and it looks at the pathogens that are causing infections during that stay. and Staph aureus is 21% of ICU infections in North America, actually higher than the other parts of the world. Why is that? Because we do a lot of things in North America because we can, because we are more resource rich. We can do more crazy transplants. We can do more complicated surgeries. We can use more complex devices. So Staph aureus, our human commensal, tends to be a real big troublemaker. And it turns out that if you have MRSA, in the next year, your chance of developing an MRSA-related infection is 1 in 7. If you are in a highly tertiary care place, it can be as high as 1 in 3. So this is not a pathogen to be ignored. Human pathogens can make us, give us trouble when you're given the opportunity. So this trial is actually not a cluster randomized trial. It is an individual randomized trial. We took about 2,100 people with MRSA. They had to have a positive culture during that hospital stay, and we randomized them on exit. On discharge, they either got CDC's general hygiene information, and we kind of beefed that up a little bit, or they got the hygiene information and they were serially decolonized. And what they got was 5 days of 4% chlorhexidine rinse-off showers, plus nasal mupirocin and a gargle with chlorhexidine. And they had to do that for 5 days, twice a month for 6 months. And what we found is when we did that, we could actually reduce MRSA infection in the post-discharge setting in the year after discharge by 30% and reduce all-cause infection by 17%. 85% of these infections cause them to come back to the hospital as a main reason for rehospitalization. So again, MRSA is not benign. It just may not be infectious while you're in the hospital watching them for 3 or 4 days. It could cause them grief much later after they've acquired it and now have risks for infection. What was really nice about this trial is we were able to show a dose response curve. If you didn't do it at all, and we had a number of people who came to our trial, came to every follow-up visit, and in every visit said, I didn't do a thing. I did none of it. And so we could compare the people who did nothing, and they were worse off than the control arm, probably because they're also not taking their diabetic meds and their antihypertensive meds and all the other things, but they were coming to our follow-up visits. Or they could do partial. And after following them for a year, they were pretty honest with us. So the people who did partial adherence, they got about a 90% benefit. The people who did, actually more than that, I can't remember the number, but it was about half, maybe 20% benefit. And the people who did it fully got a much, much greater benefit, about a 40-something% reduction in infections during that year. And this is the last trial that I will mention. We just published this trial a little while ago. This is the PROTECT trial, and now we're moving into long-term care. Long-term care in the United States is incredibly poorly resourced. And they have extreme staffing shortages, and the vast majority of the people who are taking care of patients in long-term care are nursing assistants. That means they have a high school diploma, some of them, plus about 2 months of training to put them onto the floors. So this is a trial of 28 California nursing homes, cluster randomized, 14,000 residents, and they are randomized to do what you're always doing, your routine care, your routine soap, your routine bathing frequency, or swap out chlorhexidine. Again, 4% in the shower, 2% leave on. Your usual frequency applies. We're not changing the frequency at which you're bathing. And you get nasal iodophor Monday through Friday every other week. And we use nasal iodophor instead of mupirocin. We knew at the time mupirocin was better. They just can't afford it. Even at $5 US a tube, they can't afford it. So nasal iodophor is pennies and much more affordable for them. So again, we had very typical adherence. If anything, 70% iodophor adherence. The nurse has to put it into the nose, and that can be quite challenging when most of the people on the floors are not nurses. And effectively what we found is we could reduce the reason for going to the hospital. If you get hospitalized as a nursing home resident, we can reduce the reason that it's because of infection. So 17% reduction in the reason for transfer to a hospital being infection, and a 15% reduction in the reason for discharge being that you have to go to a hospital rather than going home. And so the New England Journal said, well, that's a really interesting outcome. And we understand it was your main outcome, but that's really obtuse. Can you just give it to us straight? If I'm a resident, how much are you reducing my chance of infection? And that number is even higher. So 31% reduction in your chance of being hospitalized due to infection by this strategy. The number needed to treat is so small that effectively, when you calculate it out, we're reducing 2 hospitalizations per month per 100-bed nursing home. It's a pretty remarkable load. It also just goes to show you that in places that are poorly resourced and they don't have a lot of good infection prevention, lots of things can make things better. So overall, if you're just squinting, I don't expect you to read this slide, but it's a nice storyline because the story goes the same direction every time you tell it. So if you look at these trials, effectively what they show is that you can reduce multi-drug resistant organisms. And I'm not just talking about MRSA and VRE, which are the first ones that everybody wanted to study because this goes way back and you need something that's common. But we and others have since shown that this actually reduces ESBL, CRE bacteremia, and we'll be just about to kick off a pilot study looking at Candida auris. So nice storyline. Not only do the MDROs come down, but bloodstream infections repeatedly, trial after trial, steady as she goes, all the way through big reductions in infections and serious infections requiring hospitalizations or going to the bloodstream. So I'm now going to talk about the journey, about what happens in doing these trials. And I'll just make a few simple comments. This is HCA Healthcare that I mentioned, 200 hospitals across the United States. They have corporate governance. They have one EHR, and they have an ability to push things out, through IT practices, and it goes out to multiple hospitals. Sounds like very much like Alberta does as well. Definitely not what happens in the United States normally. So how do you decide you can do a pragmatic trial? It is really, really important that if you do a pragmatic trial, you've got to have tentacles into operations. If you don't know how things are done, if you don't know the workflow, you won't be able to really do a pragmatic trial. So, so the trials that I mentioned to you, we were fortunate that the investigative core, the content experts, all had operational links to infection prevention. They had all been leads of infection prevention in their facilities, and all of us were saying, if we wouldn't put it in our facility, we're not going to study it in a trial. Like all of us knew the limit for even wanting to study something if it was not actually going to be doable. Same thing, you want to have a system lead, health system that actually really cares about your trial. They're busy, they're running a whole bunch of stuff. And so if they're not prioritizing your trial, you're going to run into collisions because they need to run interference for a number of things, as I'll go into. So the ideal pragmatic trial has both. You have health system operators who care about your trial and are deeply engaged in the research. You've got investigators that actually understand the workflow so that they know that they're not going to cause major disruptions. You can do a pretty good job with either one of those. If you have neither, it's very unlikely that your pragmatic trial is going to go well. These were the sectors of involvement for just a bathing and nasal decolonization trial. I mean, that is all we did, right? We changed the soap and we put things in the nose, and yet we touched all of these sectors by doing it. Risk, regulatory, certainly the clinical services, nursing in a very big way. The infection preventionists were our biggest champions for signing up for this trial. Supply chain, pharmacy from Upiricin, laboratory and microbiology for the data that we needed to be able to analyze the trial, and IT also for complete data warehouse data in order to analyze the trial. This is another example about why you need corporate leadership. So if you think about hospitals, we do things all the time. You know, there's some unit who decides they can do something better, so they're going to bring something in and try it out. Someone wants to do something because there's a little bit of a fire, and so they want to throw everything at that fire, so they're going to change practice for a period of time. Quality improvement initiatives are all over hospitals, and they're in every way, shape, or form, sometimes short, sometimes long, sometimes well-designed, sometimes incredibly poorly designed. And so when we were doing our trials, we actually needed to poll every week. Please tell us if you are going to load something else out, because you have to bring it to the steering committee for us to adjudicate. We adjudicate in two ways. We either say yes or no, it conflicts with the trial. If it conflicts with the trial, meaning it has the same target output as our end goal, then you have a choice. You either drop from the trial or you don't do that competing intervention. And if you drop from the trial, luckily we had a centralized data warehouse, so we could still do an as randomized analysis. So how many competing interventions do you think hospitals have during the course of our trials? So our trials were about somewhere between 18 months to a little over 2 years during that window of time. So top one, remember ICU, middle one, non-ICU, and the bottom one, another ICU trial. So you can see these are large numbers of competing interventions, proposed interventions, competing interventions, and then because of the importance of the system, caring deeply about wanting a trial to really evaluate what we intended it to evaluate, you can see very, very few of them actually adopted the competing intervention. In the ABATE trial, those 3 adopted the other arm's intervention, even though we told them, please don't do that. So they had to drop from the trial. Cluster randomization. So I talked a little bit about why we do cluster randomized trials, but you can imagine when you think about an individual trial, like I talked to you about the CLEAR trial, 2,000 people, randomizing 2,000 people, that's a pretty good number. Cardiac trials, 40,000 people, sometimes 20,000 people. But I'm telling you about hospitals that I'm randomizing 43 hospitals, 53 hospitals, 137 hospitals. And that is a massive number of amount of work, but that is not a large number of hospitals. So how do you randomize clusters to make sure that you have balance? So we created something called the Goldilocks app. So you can actually go on Shiny, it's a free app, it's called Goldilocks, and you can throw in a number of variables that you deeply care about that's going to affect your trial. And when you do that, you can look at the universe of draws that you could potentially have when you hit that button to draw your randomized trial. So for example, the labeling is not particularly clear, but in the ABATE infection trial, in all of our trials, we said, okay, we're going to care about a couple of things. We care about the MRSA prevalence in that particular area because that's one of our targets. So if they start high, we don't want to have all the high ones in one group. We care about the volume, how big they are, because that influences statistics. And we care about how sick they are, all sorts of stuff. So we put them in and we define clinically how much difference between the arms we will tolerate. So for example, if you look at here, this is the prevalence. Does this show? Yeah. Here's the prevalence of MRSA. We would tolerate a 1.5 percentage point difference between the arms. That is all we were willing to tolerate. And we will weight that variable until all of the draws are below that threshold. So you can play with this universe of draws before you actually draw. You can get the weighting of the variables the way that you want it, and then you draw once, and then that's it. You don't get to play and redraw, but you do get to weight it the way that you want. And so this is the draw that we drew for the ABATE infection trial, a pretty good draw. This is a swap out trial, and you can see that length of stay didn't matter. It's so tight in this healthcare system that it all converges into one data point. It just doesn't matter. However, we would randomize these 137 hospitals, we got to a very, very tight length of stay. So effectively, this becomes a really useful technique to make sure that at the end of the day, you don't do all that work for the reader to say, yeah, but they're not balanced between the two arms. IT automation, this one is a big one. I think we all understand that you can do a lot more if you can do a forcing function, if you can prompt doctors to think about certain things or tell nurses that, guess what, you just have to bathe and put it on their task list so that they do it. So we created all sorts of things. Even though we were not present, we went out to train them. We were there in person to train. Other than that, everything was remote. And so these are just some of the things that we created. We have a computer-based training module, and centrally, HCA could roll it out to all nurses in an ICU or all nurses in a non-ICU. They could do it only in the sites that were participating. So overall, you can see that across a 2-year trial, we actually rolled out training that they were required to do, reaching about 9,000 nurses in year 1 and 7,000 nurses for reinforcement training in year 2 for this trial. We also created videos so that they could actually see and watch a mannequin being bathed so they could understand a bit about bathing. One of the things that's really, really important is that everybody assumes that everyone knows how to bathe and that you do not need to train on it. And that could not be further from the truth. Everything that I told you about the benefit in our trials has been shown time and time again. If you swap out product and do not train, you will get 0 benefit. I'll say that again. If you swap out the soap and you're paying a little bit more for all that chlorhexidine and you don't train, you will get zero benefit. That's been shown over and over and over. So why is that? It's because all of us know how to bathe intact skin. You grow up and you bathe yourself and you're fine. And they assume all nurses are going to be perfectly fine, except they look at their patient. They've got lines, tubes, surgical incisions, pressure ulcers. And what is that nursing assistant going to do? They're going to avoid those places because why? Because I don't know if I'm supposed to clean it. I don't know if I should touch the line. The last time I touched the line, the nurse yelled at me. The last time I touched this, the person said, that hurts a little bit. I'm not going anywhere near those places. And of course, those are the most important places to clean. Because if you don't clean the areas where the bacteria can dive, you're not going to be able to protect that patient. So we spend an inordinate amount of time in all of our trials and in my hospital every year retraining on how to bathe properly and what not to avoid, and making sure that nursing assistants and nurses really empower each other. So we do have bathing videos that go through this. This was just so funny how old healthcare is always so behind the times, right? This is like almost looks like a DOS structure, but we've all seen this kind of stuff, right? So anyways, you know, did they or did they not get a bath? Every shift has to be answered. Did they or did they not get a bath, and with what? Then we gave out adherence reports. And so if you were at one of these sites, you could drill down, you could get an automatic adherence report. You could click on it. You can see every patient, every nurse who did or didn't do that bath. You could separate it out by day versus night admissions. You could look at it by weekend versus weekday to figure out where all the losses were happening for non-compliance. And then we would feed back in huddle documents. We would literally sum it up for them and send it out every other week so they could actually tell their nurses whether or not their adherence was doing okay or not. We also had observation checklists. So it's really important, even in my hospital to this day, that you actually go out and do some peer reviews. You watch someone do a bath. So this is 10 simple yes, no, did they actually do it properly? And then some very, very simple questions. Would you feel comfortable if there was a wound? Because that patient may not have a wound, but you may not know that this CNA is like, absolutely, I will never, ever, ever touch a wound because I'm terrified. So the only way you find out is if you actually ask them and then actually retrain. In the beginning of the trial, we were doing this about 3 times a week. By the time we were done with the trial, because they won't tolerate that in workflow, 3 times a month. So, you know, you're really trying to just get them trained and then do enough to maintain so that you can provide feedback. So I'm going to talk about the unexpected, and I want to talk just a little bit. Well, I'll talk about this one. So in the very, very beginning, people said, you know, chlorhexidine can be inactivated by certain lotions. And so we actually had to have an ingredient scientist. We had to go to every hospital and take picture on picture of every tube and bottle of lotion that they stored on those shelves and then have it all adjudicated. And so I can say that it was really gratifying in 2024 when we went to look at what was being sold in the United States, and it says on the bottle, chlorhexidine compatible. So a large number of the bottles now in the United States will say, because so many hospitals are asking them if it is. The abate infection trial that I told you that it works great for people with devices, and I told you that there was an 8% non-significant difference for all non-ICUs, is actually not true. The analysis software didn't exist to do the proper analysis when we did that trial. And the proper software came out more recently where you could actually do hierarchical clustering for a proportional hazards model. And when we reran it, we were absolutely significant. And so these are things that just break your heart. Like in the beginning, you did a whole trial, you said it didn't work, you did a subset analysis, and you said, oh, and then later you say, oh, actually, it was right. The idea, though, that it's an 8% benefit is an 8% benefit. It's not a 10% benefit. It's not a 20% benefit. It's fairly small, but it's very meaningful for certain academic sites. So, for example, I went to my hospital and I said, hey, we're just about to publish this trial, and you should know that we're doing house-wide chlorhexidine bathing, and you may want to be aware that in the community hospital trial, it doesn't show that. And my leadership said, tell me how many of our patients have surgery, a multi-drug resistant organism, they have a line tube drain wound, or they're in the ICU. And of course, that was nearly everybody in our hospital. And they said, we're not giving it up. And that's actually what happened across a large number of academic medical centers, is that across the UC system from UCSF all the way down to UC San Diego, they're all house-wide chlorhexidine bathing. But for smaller community hospitals that really see kind of more bread and butter, it's probably not worth their while. And we have a cost-effectiveness analysis that goes through it. On the positive side, as I mentioned, the subset of those with lines and devices is a post hoc subset analysis. It should not make it to guidelines by our guideline criteria. It should not be widely adopted, but AHRQ, one of our federal agencies, said, nope, we're going to make a toolkit and we're going to say this is best practice. And so, like I said, practical drivers for what drives infection prevention, sometimes it's the trial, sometimes it's the setup around the trial, and the fact that everyone's driving to try to make these goalposts that continually try to reduce our device-associated infections. This one was really challenging. So we knew that chlorhexidine interacts with bleach and produces a brown stain. We've known this. We warned our hospitals when we started the trial. We never saw it, never. And ideally, we think it's because in hospitals, you send your laundry off-site. Temperatures are really high. They denature the bond between the chlorhexidine and the bleach, because they definitely use bleach, and it's a non-issue when it comes back. Day one in our nursing home trial, we saw gobs and gobs of brown stains. Nursing homes tend to have laundry on site. They do not reach the high temperatures. And our only solution, because they failed all sorts of training, because you put one wipe into you leave a wipe in the cloth and then the whole thing changes, is you have to switch from chlorine to peroxide bleach. So in nursing homes, we insist that you cannot adopt this unless you switch from chlorine to peroxide bleach, but we've never had a problem in hospitals. So I'll just end by talking about the fact that you can't adopt something without a great deal of help. And so I'm just going to fly through a couple of pictures. And so these are up. This is at AHRQ, the ICU toolkit, the non-ICU toolkit, lots of pictures about video bathing and how to do it. And then we actually put this out. So if you go to this website, we have a hospital toolkit, a nursing home toolkit, and a long-term acute care toolkit. That's the ventilator toolkit. And in each of those are all of these documents about protocols, frequently asked questions, handouts, training modules, and it's all given in editable form, because what we want is for it to be adopted. If you take the PDF version, you'll have the CDC logo and all the other logos of the people who ran the trial. If you take the Word document, you can put your own logo on it and change whatever you want just to make it more useful for your facility. It has things like this, like a one-pager, how do you actually do it? If you can't afford the wipes, how do you mix it? Our nursing homes definitely do not buy pre-ready wipes. They mix it all in order to do it. Lots of frequently asked questions that you can pull things from, wound care questions, allergy questions. You can mix and match for whatever is going on in your facility. The checklist that I mentioned. And then I'm really pleased to say that we actually launched a free resource with a e-learning module that is actually quite nice. These are these two hosted places, both NACHO hosts this, and then a California long-term care facility society also hosts it. And it's actually fairly well done. It has like a scenario. It talks you through about why these things are important. It has a video that you can play. It's got all these things about why bathing has to be taught, as I mentioned. How do you mix it if you can't buy it? How do you do clean to dirty? It's amazing what they don't know if they're not taught about the body. And then we have a mnemonic called Wash Dirt about wounds and skin folds and all these things so that they don't forget what to do when you encounter a wound or a line or a drain. And it has things about like, what do you do? This is just one of the examples of what do you do when someone refuses. And so in summary, we have large amounts of data that suggest that there are things that you can do for multi-drug resistant organisms and hospital-associated infections that can get the bulk of it into a better place. Pragmatic trials help us understand what we can expect when we adopt something that's already out there. And to do these trials requires a good amount of lift in being able to make sure you have alignment with the people who can actually make sure that it's done and tested in the way that you want. So I will stop there and thanks very much.
I think we have some time for questions, so start with Will.
Hi, thanks for the great talk. I remember in med school doing systematic reviews on MRSA decolonization and reading your paper, so it's really great to see you in person. I remember at the time, one of the complaints about the trials is that it's based in the US where at that time, certainly high rates of MRSA prevalence. So I'm wondering how much research has there been in the last few years in places where the prevalence is a bit less, say in Northern Europe?
Yeah, so they've done their own set of trials. If you take a look at the Mozart trials, they've been looking at a lot of this because they do tend to have a lot more gram negatives. I think that's why it's important to not just study MRSA, because MSSA, if you take a look at it, produces a good amount of those, 50% of infections from Staph aureus in the United States are due to MSSA. So if you look at it in combination, it is an equally virulent pathogen. I think it's not as common in those that are highly healthcare utilized. I think that those are the ones that tend to pick it up. So you do have to do a little bit of an adjustment. But what's been so important is to demonstrate that this works for the gram-negative resistant pathogens as well. So I actually think it has broad applicability, whether or not you're targeting MRSA in particular. But these hospitals, these community hospitals, they don't have a lot. And so I think it's pretty commensurate. They have about 8 to 10% if you screen. If you don't screen, it's 2 to 3%. So I think people misunderstand that they think they know their population, but if they haven't screened, they may not.
Sorry. For the targeted decolonization too, I wonder, I know we do it here in CVICU, for instance, but I'm wondering like in the outpatient setting, like with hemodialysis patients, has there been particular studies looking specifically at hemodialysis patients?
Yeah, so hemodialysis is the last trial that I really want to do. They're there for 3 hours getting dialysis. They unfortunately don't have curtains. So it's really, I mean, ideally you would bathe them there, but there's just no privacy and the staffing isn't there. So trying to see if they will wipe the arm or try to send them home. But we have shown with transplant recipients, if you send them home with a cloth to do perineal cleansing and incisional cleansing, we can get rejection to come down. So bacteriuria and rejection, so pretty dramatic. I'm happy to show you that. That's hot off the press. It'll be at ID Week this year.
First of all, I low-key want to put sitting in the sprinkler and just turn them on. But this is kind of a practical question. In my outpatient practice, if I see people on follow-up on any cases of Staph aureus, I'll routinely offer them some opportunity accommodation based on vibes. And it's probably correct. But I actually find it a practical issue with people saying, how often do I have to do this, and a drop off? And so what would your guess be? Like looking at your dose response and getting
Yeah.
yes to this spot. They usually kind of do every other month.
Yeah, I think it depends. So immediately post-hospitalization, their risk is much higher. Whatever happens that caused their initial hospitalization puts them at a high risk for rehospitalization. So that window, I would say that 1 to 3 month window is pretty important. Otherwise, it will be very skin determined. So if you've got people who have chronic skin diseases, then you need to find some sort of steady regimen to put them on. And people, there's a genetic component as well. You'll find families where the boils just run together. And then you have to do an intervention for the whole household and then try to slow it down. But secondary prevention is well described in the literature and the guidelines for Staph aureus people, and they are motivated because it's secondary prevention. They had a horrible time and they don't ever want to have that happen again.
First of all, I'll plug in and a question not directly related to your presentation, but thank you for the excellent. The plug is this, this province is, and you may already know this one of your comments, this province has a single electronic health record for the entire acute care system, 6 hospitals, and 1 single lab system. So you're looking to expand beyond the US, perhaps into that.
Yeah, you are ripe to do some really nice trials. Yeah. Yes. Happy to, we'd be excited if you'd like to do the hemodialysis trial or something else, please let me know. There's so much to be done and so much that you can do.
Question.
may or may not be fair, and certainly isn't directly related to your talk, but those of us who follow these things are usually concerned about what's happening to HICPAC in the US and what your administration has planned or.
So HICPAC, for those of you who don't know, is the guidance committee that provides all of our infection prevention guidance from the United States ever since the 1970s until today. HICPAC is dead. It has been disbanded and destroyed as of 2 weeks ago. And so we are in a grave problem with our administration. Their view of health is really antithetical to what we believe needs to be done and needs to be protected. So I think we're in a lot of trouble. You can see it in the news, the vaccines, we're in trouble. So I think that the talk that was given about information and trying to hold out to try to persuade people who believe something, believe that they're correct, and believe that that's the right thing for health, and yet we're not making inroads. And as an infectious diseases group, we're not making inroads. Our societies are having a hard time. We're just trying to get healthcare workers to have access to the vaccine at this point. Like, we don't know if we'll be one of those protected groups. Otherwise, you might see me back here getting a vaccine. So yeah, so I wish I knew. It's been awful. Grants, yeah, it's been an absolute catastrophic destruction of research for health. On that sad note.
Well, I will not stand in the way of your lunch. Please help me in thanking Dr. Susan Huang, who's and we do have lunch just outside the doors. So you can get your lunch, you can come back in here and eat it. I realize this is not ideal. It's a big group, but this is what we have to work with. And then if you do have questions for any of the speakers today, please go ahead and ask them. And thank you very much for coming.